We now move on to our third session titled Basics of Pain, Neurobiology and Pharmacology, a critical topic in understanding the mechanism of pain and its effective management. This session will be led by our esteemed speaker, Dr. Rajan Radhakrishnan, Professor of Pharmacology at College of Medicine, Mohammed bin Rashid University of Medicine and Health Sciences, Dubai. Dr. Radhakrishnan is a renowned expert in this field with a significant contributions to pain, research and pharmacology. To chair this session, we have a privilege of welcoming Dr. Anil Babu, a principal of Westford College of Pharmacy, Thrissur. Both are requested to take their position on the dais. It's an absolute privilege to introduce Dr. Anil Babu, a distinguished academician in pharmaceutical sciences. Sir has over 17 years of comprehensive experience in teaching, research and administration. He has published 39 research papers in reputed journals, supervised 20 theses and actively contributed to clinical and public health research. Dr. Anil Babu is actively involved in scientific activities, having participated in and organized numerous conferences, workshops, medical camps, and serves as reviewer for leading international journals. With due respect and honor, I invite Sir to chair this session. Sir, please.
Good morning, everyone. First, let me express my sincere gratitude to the Holy Grace Academy as well as the management and director and other faculty members who has given me an opportunity to be a part of this Hapscon 2025. It's really an honor and privilege to me to introduce today's esteemed speaker, Dr. Rajan Radhakrishnan, a highly respected expert in pharmacology and biomedical science. Dr. Rajan Radhakrishnan is currently a professor of pharmacology at the College of Medicine at Mohammed bin Rashid University of Medicine and Health Science in Dubai. He also serves as a chair of Biomedical Science Graduate Studies Steering Committee, overseeing key aspects of MSc and PhD programs in biomedical sciences. Dr. Rajan Radhakrishnan's educational journey is quite impressive, having earned his PhD in pharmacology from National University of Singapore, after which he completed a postdoctoral fellowship in pain neurobiology at the University of Iowa. His academic foundations also include an MSc in pharmacology from University of Strathclyde, UK, and a BPharm from University of Kerala, India. His professional career spans over several decades with key roles in both academic and research leadership. Notably, Dr. Rajan Radhakrishnan directed the master's program in biomedical sciences at Mohammed bin Rashid University, where he oversaw the curriculum development, student progression and admissions, contributing to the high standards of the program. Dr. Rajan Radhakrishnan's editorial roles underscore his authority in the field. He serves as the editor of phytotherapy research and has held editorial positions in journals like Inflammatory Pharmacology and the Journal of Pain. Additionally, he has been a reviewer of more than 16 prestigious journals. Throughout his career, Dr. Rajan Radhakrishnan has authored over 50 publications and contributed to various book charters. His research and teaching continue to influence the field of pharmacology and pain neurobiology, providing valuable insights into the therapeutic strategies for managing cardiovascular diseases, inflammation and pain. It's an absolute privilege for me to welcome Dr. Rajan Radhakrishnan, sir, for his wonderful sessions. Sir, please, stage is yours. Thank you, sir.
Thank you very much, Anil, for the kind introduction. Good afternoon. I know that you'll be very tired after these many sessions, so hopefully you will keep awake and keep quiet, if possible, a little bit, so that you can listen to me. I was planning to give an hour talk, 1 hour talk, but I will try to cut it down to 30 minutes so that you guys can go and have your lunch break. So my topic today is pain, neurobiology and pharmacology. My area of research is pain, but I decided not to give a research talk today because it'll be too detailed for you and probably boring for you. So what I will do is, I will try to give an overview of pain neurobiology and also will try to explain how drugs they act to reduce pain at the various sites of pain pathway. So that way I think it will be more connected to you and what you need to know. And as I go through the slides, I will try to connect these things with my research area. So it will be in context with what you need to know and what will be relevant to you. Okay. So these are the learning objectives for my talk today. And usually when you get a lecture, you will get these learning objectives. Most of you don't read through the learning objectives, even when the professor asks you to read through them. So it is extremely important to know the learning objectives before you start listening to a talk or a lecture. So these are the general learning objectives for today's talk. Basically to understand the pain mechanisms, how pain is transmitted, and how we can modulate or how we can manipulate transmission of pain so that you will get pain relief or analgesia, right? So maybe students in your third or fourth year must have learned pharmacology of pain or neuropharmacology in general, which includes pain pharmacology. So you will be more familiar with the pain mechanisms. And also I will try to classify analgesic drugs very briefly because as I mentioned to you, the time is very limited, only 30 minutes. I'll try to finish by 12:45. And then being pharmacists, the last point is very important, suggesting an appropriate analgesic agent for a given patient based on the patient's specific information. So this is what we need to learn and what we need to practice. And you should be experts in that last point, being pharmacists. Although the system in India has not developed to a point where a physician will ask a pharmacist to suggest a drug, but that will come in the future. Professor Jayashekhar is here. He's leading the IPA initiatives in Kerala. We were talking about this particular point that we as pharmacists need to get into the actual patient care. Like Dr. Erin was telling the same thing. In the US, it is very common that a physician will consult a pharmacist before they prescribe. Sometimes even to calculate the dose, especially in inpatient settings. They will check with the pharmacist. The pharmacist will help the physician to determine the dose. So we should get to that point eventually. I know that it is difficult, but unfortunately in our country we need a lot of political push for those things to happen. It has to be mandated by the government, and then slowly the physicians will start listening to the pharmacists. But that is one point. But the main thing is that we as pharmacists should be experts, should know things to advise the physician. So that is important too. So we cannot just ask for the physicians to consult us, but we should be capable of advising them, and that is more important. So you should be knowledgeable about these drugs, about the mechanisms, dose, drug interactions, and so on. And that point will come maybe in another, 10 years, hopefully. So pain, this is the definition of pain. It is an unpleasant sensory and emotional experience associated with actual or potential tissue damage. So that is the accepted definition of pain. And there are these terminologies about pain which I will skip through: allodynia, hyperalgesia, pain threshold. I think you are all familiar with pain threshold. Analgesia, that is more important. Analgesia is the inability to feel pain while you are still conscious. So it's the difference between analgesia and anesthesia is that in most forms of anesthesia, especially general anesthesia, your consciousness will be inhibited. And of course the pain will be inhibited, but with a good analgesic, it will selectively inhibit pain. So that is the definition of analgesia, and analgesic is an agent that will suppress pain. How many of you are familiar with this pain pathway? I think in physiology you must have learned about pain pathway. Anyone familiar with this pain pathway? How pain is transmitted from the periphery or the site of origin of pain to the higher centers of the brain where it is perceived as pain. So that is the whole pain pathway. And being pharmacologists and pharmacists, we need to know the pain pathway so that we can describe the mechanism of action of analgesics. Because we learn about analgesics, we memorize things, but we don't try to understand where actually these drugs they act in the pain pathway. So let me see if I have a pointer here. Oh, sorry. I don't think I have a pointer. So if you look at location 1, that is the peripheral nerve terminal. So those nerve terminals are located in the tissues in the periphery. So those nerve endings, they have sensors for pain. So they will sense pain from them, from the tissues, and transduce it, make into electrical current, and that electrical current will be transmitted through the neuron. And that site number 3 is the spinal dorsal horn. So it reaches there and it will communicate with the second order neuron you can see at site 3, which will take the pain signals to the brain. So that is where you perceive pain, mainly in the cortical areas. That's where pain perception happens. So why is this important for us, pharmacologists and pharmacists? For example, NSAIDs, we are all familiar with NSAIDs, right? Non-steroidal anti-inflammatory drugs. Ibuprofen, diclofenac, indomethacin, aspirin, all those drugs. So we call them collectively as NSAIDs, non-steroidal anti-inflammatory agents. So that site 1, if there is an inflammation in the peripheral tissue, let's say muscle, skeletal muscle, we have an inflammation. So at the site of inflammation, these peripheral nerve endings will sense pain from those inflamed areas. That's why we feel pain when there is inflammation. And one of the mediators of pain and inflammation is prostaglandins. There are several types of prostaglandins. So these NSAID drugs, non-steroidal anti-inflammatory drugs, they inhibit the synthesis of these prostaglandins and reduce the levels of prostaglandins. Therefore, you feel less pain, less inflammation. So that is one mechanism of reducing pain in the periphery. So this is how NSAIDs they act, right? Non-steroidal anti-inflammatory drugs. I'm sure that you learned the pharmacology of NSAIDs in second or third year. Okay, so that is one way of reducing pain. So when the pain signals come to the spinal dorsal horn at the site number 3, transmission happens between primary afferents and the second order neurons going to the brain. So drugs like opioids, I'm sure you're familiar with opioids like morphine, fentanyl, oxycodone, hydrocodone, all those opioid drugs. They reduce the release of neurotransmitters from the primary afferents so that the signals coming on the primary afferents will not be transmitted to the second order neurons. So this is the site of action for opioids, site number 3. So we already covered 2 major painkillers, right? NSAIDs, opioids. What is the other common analgesic agent? Paracetamol or acetaminophen. Acetaminophen is the US name, paracetamol is the Indian name and British name. British territory, they call it as paracetamol. So where does paracetamol act? Does anyone know the mechanism of action of pain relief by paracetamol? Is it an NSAID? Is paracetamol a non-steroidal anti-inflammatory agent? No. No, right? It is not considered an NSAID because paracetamol has no anti-inflammatory activity. There is a theory that it suppresses COX-3 in the brain, but does not have any peripheral anti-inflammatory activity. So it is not generally considered as an NSAID. Okay? So there are several theories behind the mechanism of action of paracetamol. One says that it has some opioid-like effect. Another theory says it inhibits the reuptake of monoamines, like the antidepressants. That is one theory. And the other theory is central COX-3 inhibition. So those are some of the theories, but we don't know for sure what is the actual mechanism of acetaminophen pain relief. So anyways, I think we covered 3 major classes of analgesic agents that we use commonly in the clinical practice. So my whole lecture is about those drugs. So I think with this one slide, we have covered half or more of my lecture. And especially with the time constraints, I think the slides are not moving. Maybe I should use this, okay. So pain processing, I tried to describe what I just mentioned to you. So that's, and these are the pain carrying fibers. A delta fibers carry sharp pain when you get a pin prick. The pain signal or message is carried by A delta fibers. Dull aching kind of pain, like muscle pain you feel in the shoulders or muscles, is carried by C fibers. So this is a pain classification, nociceptive pain, where there is a cause for the pain, in a peripheral cause. Post-operative pain, inflammatory pain, they all come under nociceptive type of pain. The other type of pain is neuropathic pain. Neuropathic pain you mostly see when you practice. You see this with diabetics or diabetes patients. That is because of the neuronal destruction that happens in diabetics. So diabetic neuropathy is very common. Other types of pain, referred pain and fibromyalgia. So referred pain does not fall into these 2 major categories. Fibromyalgia, as some of you know, it does not fall into either inflammatory or nociceptive or neuropathic pain. We don't exactly know what causes pain. There are several theories again behind fibromyalgia. So nociceptive pain, this is how pain signals are conducted, goes to the spinal cord and then to the brain. So mainly we use prostaglandin inhibitors, opioids to treat nociceptive pain. Neuropathic pain is mainly caused by nerve injury, whether it is peripheral or central neuronal damage. And the traditional analgesic agents like acetaminophen, NSAIDs, they don't work in neuropathic pain. So what they do is they use sodium channel blockers that we use as anticonvulsants. That is one option. And then the other option is antidepressants. So they use antidepressants to treat neuropathic pain. Opioids are effective, but of course opioids, they have the addiction potential, so we don't want to use opioids for chronic type of pains in general. So we don't prefer opioids in treating neuropathic pain. So story about the pathophysiology of neuropathic pain and all that, referred pain, I think you're familiar with the heart attacks. People with heart attack, they get pain in the left hand, left side of the body. Actually, there is nothing wrong with the left side of your body. There is no ischemia. Ischemia is in the heart muscles, but it is referred to the left side of your body because the afferent fibers going from the left side of your body and the cardiac muscles, they converge at the same point in the spinal cord. And the brain interprets the pain as if it is coming from left side of your body. There is nothing wrong with the left side of your body. There is no ischemia. So that is referred pain. And you see these with other organs like kidney, liver, etc. When you get pain, you feel pain in other areas. Fibromyalgia. So one major theory behind fibromyalgia at this point is that it is a pain that occurs or originates in the central nervous system. There is nothing wrong. You can see if you do an MRI or a CT scan, whatever imaging you do in the periphery, you don't see any problems. No abnormalities in the periphery. So it is a central augmentation of sensory input and deficits in central inhibitory mechanisms. So that is the latest theory behind fibromyalgia pain. Drugs approved, we have pregabalin, duloxetine, milnacipran, and those who are familiar with the pharmacology, they know pregabalin is an anticonvulsant drug, but very useful in treating neuropathic pain. Duloxetine, again, is an antidepressant. Milnacipran, another SNRI antidepressant. So these are used in treating fibromyalgia. Now, we have seen the neurobiology of pain very briefly. You know how pain is transmitted. So coming to the pharmacology, you have two major types of analgesic agents. One is traditional analgesic agents. We already mentioned about those, like NSAIDs, paracetamol, opioids. So they are all traditional analgesic agents. We have non-traditional agents such as sodium channel blockers, antidepressants, those type of things. So those are not usually used for treating, nociceptive type of pain. For example, you get headache, you don't take anticonvulsant, right? So these are non-traditional painkillers that are used mainly to treat neuropathic type of pain. WHO pain ladder, again, in the context of pain medications, is good to know, especially in cancer pain. This is the order you go with treating cancer pain. Start with non-opioids and some adjuncts, non-pharmacological adjuncts. If the pain is not relieved, then we go to non-opioid, again, plus or minus, maybe sometimes you add an opioid. And then for severe pain, you opt for opioids. So you go in a step-like manner. So for non-neuropathic pain, these are the classes of drugs. We have steroidal anti-inflammatory agents. We don't usually use for treating pain. But if there is inflammation involved, then we sometimes use steroidal anti-inflammatory agents, right? Like you have prednisone pack, sometimes you use for 7 days for severe inflammation. NSAIDs very commonly used for treating pain, especially inflammatory pain. Opioids in moderate to severe pain, especially in post-op hospital settings, we use opioids. There are miscellaneous drugs which we cannot include in any single class, like acetaminophen. There is no specific mechanism. Tramadol, I think you have studied in pharmacology. Tramadol is a mixed mechanism drug. It has some opioid effect, some antidepressant type of effect. And then tapentadol is very similar to tramadol. again has strong opioid action for tapentadol. So tramadol and tapentadol, they are controlled substances, controlled drugs. Again, I think I will skip through these slides because we don't have time. Commonly used NSAIDs, and you're familiar with these NSAIDs, right? Most of these, especially the blue ones and the green ones, very commonly prescribed. The red color one, ketorolac, you may not be familiar, but it's a very useful NSAID, usually used in hospital settings. It's very useful for severe pain, moderate to severe pain. That is the only NSAID that is useful in moderate to severe pain. Other NSAIDs, if you look at the indication, they're mild to moderate pain, right? NSAIDs are generally acetaminophen also, mild to moderate. But ketorolac is the only NSAID that is very useful in moderate to severe pain, and it is usually used in ER settings or emergency room settings. This one I think I need to mention. When you talk about NSAIDs, there are two types of NSAIDs you should know, right? Selective and non-selective. Most of the agents we dispense, they are non-selective agents, ibuprofen, diclofenac, indomethacin, naproxen, all those are non-selective. Selective ones are very few. Celecoxib is the common one, Celebrex, the brand name. So that is one of the common ones. So what is the advantage of selective COX-2 inhibitors? Those who have learned pharmacology of NSAIDs should know. What is the advantage of COX-2 selective inhibitors? They are gentle on the GI tract, right? It does not cause any GI upset, ulcers, bleeding, etc. It's not caused by, at least in theory, it should not cause GI issues. So, celecoxib is the one that is very commonly used. There are several drugs that were withdrawn from the market because this class of drugs is a problem. It increases the cardiovascular risk. It increases the incidence of cardiovascular diseases in patients who are taking it for a longer period of time or chronic use. Are they superior to traditional NSAIDs? When it comes to clinical effect, which is anti-inflammatory effect, it does not have any superiority. But when it comes to the ADR or adverse drug reaction, then this one is superior to traditional NSAIDs. Because those who are sensitive to NSAIDs, especially if their GI is sensitive to NSAIDs, then this class of drugs will be useful for them. And these are the general cautions we should observe when you dispense NSAIDs. So again, I mean, these are all very general information, right? So I tried to put these in context with the pain neurobiology, which you may not be familiar with. So now probably you will be able to better understand the mechanism of action of these analgesic agents when you learn about them in the context of the physiology, right? So again, with the COX inhibitors, there is another issue, which is possibility of bleeding, right? Non-selective NSAIDs can cause bleeding, or it can prevent platelet aggregation. And I think you're all familiar with that because we advise patients to take aspirin to prevent heart attack. We're all familiar with that, right? And the reason is that non-selective NSAIDs, they also inhibit COX-1 enzyme. COX-1 enzyme produces what? Thromboxane A2, which causes platelet aggregation and clot formation. So if you take aspirin, you can deactivate platelets, so they will not be able to form aggregates, clots, and that can prevent heart attack. So some people, they like to take NSAIDs before they go to a dentist, so that preemptive prevention of inflammation. So this is again like critical thinking question for pharmacologists. Which type of COX inhibitor should you suggest to a patient going for dental extraction so that they will get minimal amount of bleeding? Should it be non-selective or selective? So it is ideal to take a selective COX-2 inhibitor so that you are getting rid of the COX-1 inhibitory activity, which will reduce the possibility of bleeding. So again, for those who have taken pharmacology course, I think they will be better able to appreciate these points. Acetaminophen, I think you know everything about acetaminophen or paracetamol. What are the theories behind its effect? And not to exceed 4 grams a day. I think we all should know that, right? You all know that, I hope. We should not exceed 4 grams per day in an adult. It's very important. And the most important toxicity is hepatotoxicity. As you know, IV formulation available. And in overdoses, what do? So there is a specific antidote for acetaminophen poisoning, which is N-acetylcysteine. So that is given either orally or IV to treat overdose. So again, my question, so it is not generally considered as an NSAID. Yeah? So even some physicians, they think that it is an NSAID. So we should try to correct that. So I think I will try to wind up just to show the number of deaths caused by acetaminophen alone in the US each year. About 300 people die of paracetamol, which we consider as a safe drug. And it is usually due to unintentional overdose. So although we consider it as a safe drug, we should be careful with acetaminophen. So how do you treat acetaminophen? Opioids, so these are some of the common opioids that are prescribed. So when you start dispensing or you practice in hospital pharmacy, you will come across some of these opioids. So I think I will probably stop here because of the time constraints. If anyone needs the slides, I can send the slides to you if you want to read in your spare time after your exams, maybe when you are free more in the summer time. So thank you very much for listening, and thank you again for inviting me to give this presentation. It was very nice to see you all. Very good participation. Congratulations to the administration, Dr. Ajit Kumar, for organizing this wonderful event. Thank you very much.
Thank you very much, sir, for having a brief note as well as sharing your professional expertise for today's audience. Without wasting much time, we can go for the QA sessions. If anyone have any kind of doubts, you can share with sir. Is there any questions regarding? I think we can wind up the session, sir. On behalf of everyone who gathered over here, we thank you, and we take this opportunity to really appreciate your effort you have put forward over here to share your knowledge. So thank you very much, sir. Thank you.
It was an honor to have Professor Anil Babu sir and Dr. Rajan Radhakrishnan sir. As we would like to take a moment to present Dr. Rajan Radhakrishnan sir with a token of gratitude. For this, we would like to call upon Mr. C.V. Jose sir, Finance Director, Holy Grace Group of Institutions. Sir, please. Now, I also request Mr. Jojo sir, Finance Head, Holy Grace Group of Institution, to present our token of gratitude to Dr. Anil Babu Sir, Principal, Westford College of Pharmacy. Sir, please.
Thank you, sir. Further, we are moving on to the fourth session of today's conference, and this session is dedicated to the topic Evolution and Current Trends of Regulatory Affairs in Pharmaceutical Industry. We are privileged to have the eminent speaker, Dr. L. Ramanathan, Associate Vice President, Technical Operations, Apex Laboratories Private Limited, Chennai, and Chairperson, Professor Dr. Shyam Kumar, Principal, Al-Azhar College of Pharmacy, Thodupuzha, with us to guide this session. With due respect, I request them to occupy their seats on the dais. I request the delegates to remain silent during the sessions. To chair the session, we have the esteemed Professor Dr. Shyam Kumar B., a distinguished academician and researcher, currently serves as the principal of Al-Azhar College of Pharmacy, Thodupuzha, with over 20 years of teaching and administrative experience in the field of pharmaceutical chemistry. He holds a PhD in pharmaceutical sciences from Niti University, which he conducted groundbreaking research on the pharmacological and phytochemical properties of medicinal plants. His extensive contributions include publishing over 12 peer-reviewed articles in reputed journals and presenting papers at national and international conferences. His vast expertise, commitment to innovation and leadership qualities make him a highly respected figure in the pharmaceutical sciences community. We are deeply honored to have him to chair this session. Over to you, sir.
Hello, very good afternoon. This is pleasure to come to Holy Grace Institution. This is a homecoming for me. First, I express my gratitude for that. And moving to today's session, that is evolution and current trends of regulatory affairs in the pharmaceutical industry. Today we have with me my esteemed speaker and resource person, Dr. L. Ramanathan sir, a distinguished professional with a wealth of experience in technical operations, regulatory affairs and innovative pharmaceutical research. Dr. L. Ramanathan is currently serving as the Associate Vice President of Technical Operations, Apex Laboratories Private Limited, Chennai. In his role, he handles a wide range of responsibilities, including overseeing regulated markets, clinical trials, pharmacovigilance, and the development of innovative research and development products. His expertise in navigating complex regulatory process is evident from his attendance at key meetings such as pre-IND meeting with US FDA and the scientific advice meeting with Federal Institute of Drugs and Medical Devices, Germany. His academic qualifications include a Master of Pharmacy from JSS College of Pharmacy, Ooty, where he graduated with a distinction, and PhD from Mathura Kamaraj University in collaboration with Israel Institute of Technology. Throughout his career, Dr. L. Ramanathan has earned several prestigious awards, including UGC JRF and UGC SRF for meritorious students, as well as Israel Government Scholarship for his contribution to a European Union framework project focused on nanocomplexes for bioactive agent delivery. His impressive innovation capabilities are evident with several patents, including a US patent for skin disorder treatments. In addition to his role at Apex Laboratories, Dr. L. Ramanathan has been deeply involved in several pharmaceutical organizations. He currently serves as the Vice President of Tamil Nadu Pharmaceutical Manufacturers Association, Joint Secretary of Confederation of Indian Pharmaceutical Industry, and various leadership roles in other notable associations. His contribution also extends to academia, as he has served as a resource person at the College of Pharmacy, JSS University, and as an expert member for the Board of Studies at SRM University. Dr. L. Ramanathan has also published 8 international research papers reflecting his strong academic background and commitment to advancing the pharmaceutical sciences. His areas of expertise range from regulatory market fillings for the US FDA and European Union to the development of innovative pharmaceutical products. So with these words, I welcome Dr. L. Ramanathan and hand over to you, sir, for this session. Thank you.
Good afternoon, everyone. I didn't hear. Good afternoon. Thank you. I stand between you and lunch. So if you are cooperative, we can finish off and go to lunch very fast. Right? Thank you. So at the outset, I thank the Holy Grace Management for this international conference and also our ever enthusiastic Jayasekhar sir. Whenever he calls, I can't say no. Looking at his age and enthusiasm, I envy of him always. And second thing, I thank all the fellow pharmacists who has come in large numbers. I know it will be too much for a day, but still no. A conference is what he said. Very important point is the interactions and the liaising, which is going to take us into larger heights. So with this, I'll just move on to my presentation. So just a brief introduction about my company, where I represent, just one slide. So for your information, I am third generation pharmacist. My grandfather, my father, and I am the third generation. So pharmacy is not a very bad profession. So it's a really earning profession. And about the company, my father was an entrepreneur and he was a group director in Apex Laboratories, Chennai. Most of you will know about the product. Have you heard about Zincovit? Yeah. So Zincovit is one of our brands, and we also have paracetamol P group, which is P125. It's around 16 variants. So we are number 2 in the India. So we are from Chennai basically. We have around 4 facilities, manufacturing facilities in and around Chennai, and we do have our own in-house R&D. So that is one of our strengths. So we host around 100 full-time scientists, so who is working around the clock. So we do have around 45 international patents. So this year alone, my team has submitted 5 international patents, and being a regional company, we have filed NDA. So probably I'll take you over there. So now regulatory is a very, it's kind of a boring subject. So to make it little easier, we'll start with the dessert. So can you say a difference between these 2 desserts? Can someone? Can someone know? Anyone? What is the first one and what is the second one? Can somebody speak in mic, so that every day we take, right? Maybe afternoon also we might take. So anyone? Just a guess. See, if you guess and probably we can get a chance for a plant visit, maybe a training or whatever. No, you can just try. Faculties also, no one? So, first one is the ice cream. Second one is a frozen dessert. A frozen dessert consists of vegetable oils. This one is fully milk. So, how important regulation is, right? Now we understand. Every day as a pharmacist, as a general being we take. So, we'll see a couple of examples. You can see this. There is a point that not recommended for children. Do you know why? Anybody? Wild guess? Not recommended for children. We drink daily. We don't see the label. We say label for 2 things, right? One is for the price. Second is for the manufacturing and expiry date, right? It's written not recommended for children. because it contains artificial sweeteners. Any artificial sweeteners, saccharin, neotame, aspartame, whatever is there, it is not recommended for children. So there are regulations. And third thing, you know difference about this, ORS, and also lot of juices will be there in FSSAI products. When you go to the shelf, it will be like electrolyte, it will be in different names. What is the difference between a ORS and a general product, FSSAI product? Can anyone tell? Everybody takes ORS, right? What is ORS? Oral rehydration. Okay. What it contains? It's a WHO formula. Sodium, potassium, glucose. Right? That is if you follow the ORS of WHO, it is ORS. Other products which are all other than ORS, nobody can claim that. Particularly FSSAI product which is in fruit shape and everything, you can't claim that. And important parameters, whenever you look into FSSAI, see, we have drug, we have FSSAI, we have so many things in that. FSSAI is something which is directly between the consumer. It is kind of there is no doctor involved, there is no nutrition, right? Maybe nutrition, dietitian may be involved, but you go and pick from the shelf, right? When you go to a shelf, you pick that. So whenever you pick a product, we need to see the principal display panel. What is the principal display panel? Not the price, not the thing. They will give all the informations on that. So whenever there is an information, we have to read that. Some cases it may be soy allergy has to be mentioned, milk, somebody will have milk allergens, lactose intolerance. So everything has to be displayed. So this kind of regulations are there. So before me, the other speaker was talking about paracetamol. Mostly it's an antipyretic drug. You know what is the combination strength approved in India? Anybody? Paracetamol, there are a lot of strengths, up to 650, 500, 125, 100, 250, right. In combination, what is the approved limit the government has given? Anybody? It's 325 milligrams. If paracetamol is combined with any other drug, it should not be more than 325 milligrams. If paracetamol is taken alone, it's fine, 650, up to 650 it's allowed. But when you are combining paracetamol with any other drug, it has to be 325. So this is just a vagueness. So now I'll try to explain my situation, my company's process in the regulation as well as just to give you an impact on that. So we all know these regulatory bodies, right? These many bodies are involved in a pharmacist's life. So you have FSSAI, you have price control, NAPA, they control the price. You have national list of essential medicines. Medicines are always considered as essential commodities. So they will frame a list and they will say this is the price you have to keep. Initially the DPCO was like based on the raw material input. What is the price of raw material? That is the final product price. But now they have made a MAT based on MAT. So NAPA is very interesting area. So you can relate to which profession you want, which area, because whenever we talk about pharmacy, people will think that I go to production, I go to R&D, I go to QC, okay. What in R&D? Do you know how many roles in R&D is there? Anybody? No. So its formulation team is there, analytical team is there, microbiology team is there, DQA, documentation quality assurance team is there, tech transfer team is there. So it's not that nobody wants, like I have a resume that says that I fit into all profiles, international standards. So we need to understand what are all the segments. So this is one of the area, even pricing, CDS, of course, you have national agency, narcotics. So all the controlled substance, psychotropic substance falls under the CBN, and then you have IPC monographs, then you have international agencies, FDA and all. This ICH, right? Whenever you work on the formulation, see, there are many, have you thought about it? I have thought a lot. We have Indian standards, we have Europe standards, we have US standards, we have Japan standards. What is a common standard? So that is the thing you have to look into ICH. Whenever you are making a project, maybe B.Pharm, maybe a final year project, formulate as per the ICH guidelines. it has given for everything, quality and all the things. So based on which you have to go. Again, regulatory affairs, I'll tell you, it is a serious profession now, because earlier it is like, I file for a license application, that's it. But it's now it has become a lot of information to the development as well as end product. So you have combination products, strategic development, you have accelerated pathways. So I'll talk about it. The regulatory agency interactions, submissions, compliance, now medical device again. So you can choose among that. See, when you are doing B.Pharm project, final year project, always don't take whatever our seniors have done, whatever in library it's available. Just go for a new product, new project, kind of in regulatory, whatever guidelines or anything, which might be useful for us as well as for the juniors. So now earlier it was like regulatory data, now it's about regulatory decision. So we work in cross-functional teams. See, the company is completely systemized. Now you can't say that everything is auditable. For example, I have a product developed in the R&D that has to come into the plant. So scale-up batch has to be taken. For the documentation control will be there. For that they will be making a change controls. So that change control, which team will be monitoring? Quality assurance. So quality assurance is one of the key areas wherein you can select your future. M.Pharm quality assurance is there, M.Pharm regulatory affairs is there, not only M.Pharm Chutics, but a lot of areas are there. So earlier, as I said, earlier it was like only regulation was more and science was less. Now, after all this COVID and everything, the regulation has become less and the science has become a lot. So we have a lot of things to work on. So particularly after AA and all, of course we also work with AA. I'll just brief about that thing. So we know the evolution. FDA was the initial one, and then after ICH was there, and then European medicines, and then CDSCO, European EMA agency. Now earlier UK MHRA was there, and then now it has been there. After Brexit, it will be completely EMA. So if you work on any project, you think about which market you are going to work, either for regulated market, domestic market, or ROW markets. Main regulators will be US FDA, Brazil Anivasa, Mexico, South Africa, Swissmedic. So in Europe they work in a different style. EMA, like there are around 26, 27 countries, right? So you can go for a common application, or you can have a split application as well as reference member state and then concerned member state. You can apply for one country and then take it to another country, or you can go for a common product, but that product has to be novel and it will be a full mixed application. So this is the one I was talking about, ICH. So if you understand ICH, it talks about quality, safety, efficacy and multidisciplinary. So this guideline will be kind of a Bible, Quran, whatever you say in a Gita or for us. Other than Drugs and Cosmetics Act, we have to understand this. This is where the whole world is towards. So I will talk about our experience. Morning they were talking about NDA and all. Yes, Indian companies are generic companies, I understand. We are a regional company. We are around like 1,000 crores. We are among top 40 corporates in India. But still, we invest in NDAs. So in NDA, it will be like preclinical, IND submission, clinical, NDA submission, FDA review. Yeah, this is a common thing. So there is a time period for that. So when you go as an NCA route, you can have a lot of options. Next slide, I will just tell you the phases. So in this, I will broadly tell you. For example, our product is an topical product. So morning somebody asked about the antimicrobial resistance. So antimicrobial resistance, again, US will be very strict. They have a system for that. So there is a study, kind of vancomycin study model, the passage study they will say. So you can see that how much is the resistance been developed over the product. If you don't have the real-time data, that study can be used. Like they will do the minimum inhibitory concentration as well as the passage, and they will study in all strains like clinical isolates, the toughest strains which our product develops resistance over the period of time. So all these studies, concepts has to be developed, and then before IND, US FDA gives a chance for you to apply for a pre-IND meeting. Here in India, it's, of course, there are a lot of improvements, but you can't directly talk to a regulator and understand things. But there, over a cup of coffee, you can file the application, you can ask them which pathway is suitable for my product. And mostly, CDAR meeting, there were around 20 people, and half the crowd was from India. One madam was heading, she's also from India. So, people are willing to help you, they will guide you the strategy. You can ask a lot of questions. So once upon that, you can fill your preclinical study based on that. If you are developing a preclinical, you have to study on the toxicity profile. There will be more on toxicity, genotoxicity, mutagenicity, and then activity model also. As he said, no, rodent, as well as they will be asking for a non-rodent species. So non-rodent, usually mini pig is the preferred model. You can go up to higher animals. We have done a porcine model also. So based on the reports, you have to file an IND. So in IND also you can have a questions, written questions. So once the IND is, now we are in IND stage, we have done our preclinicals. So we are waiting for a phase 2B study. So this month I will be going to US for evaluating the CRO. Yes, of course, it will be like 30-40 crores. As of now, our company itself is funding for this particular project up to phase 2A. But the returns in US, more than ANDAs, NDAs is huge. And then once phase 2 is good, phase 2B study is also there an option, or you can go for phase 3. So phase 3 is a large number. As morning he was saying about clinical trial, there are a lot of avenues. For example, have you heard about orphan diseases? Orphan diseases is something which is very rare in population. India, now only they have classified, they have made a word called orphan disease. In US and EU, they have a lot of orphan diseases which is called as rare diseases. So these rare diseases comes under fast track policy. So you can fast track your applications. So this trial, of course, there will be a registry, there will be agency, everything, but you can file through that route. So we are filing a rare disease, an orphan condition. So once that is done, then the phase 4 will be there. Of course, phase 4 MedWatch is there, PDFA is there. Nowadays, India also they have brought in pharmacovigilance. So there is not a big thing. They will be monitoring cell signals, whatever the product is triggering any adverse reaction, critical adverse reactions. That they will be monitoring, and then they will have a review meeting. Then you have to submit the NDA application. Then it will be reviewed, and then drug labeling will be done. In the meantime, your facility also gets triggered. Once you file an IND, your facility gets triggered for an facility application. So that is the fast track approval. I talked about fast track approvals. So this is our own experience. It is very, very, very okay, but it takes time, but it is very much possible. In India, we know that a lot of changes have occurred after COVID. Particularly, the agency has approved fast track approvals. The hydroxyquinone cases were there. They have now amended New Drug Clinical Trial Act 2019. Audio video recording. See, clinical trial, if you think that Form D students particularly, clinical trial is one of the avenue which is going to go to the next level, because even for nutraceuticals, people are asking for scientific rationales. So we have a clinical trial inbuilt team. So what we do is we work with CROs for site identification, site initiation, site monitoring, all the statistical data. So a lot of things can be done in clinical trial areas. Of course, we are almost contributing to 20% of the world population through the vaccines developed from India, as well as we have supplied these hydroxyquinines. So naturally, we are generic pharmacy of the world. Every third pill which is consumed in the world is made in India. Okay. Now that how pharmacovigilance arrived was like, there was an incident, thalidomide, thalidomide, it had a phocomelia kind of side effects to children. You can see the shortening of limbs. After that only, FDA and everybody took the seriousness of this is a medicine. Of course, it's not only developed and has to check for quality, but it has to monitor for the life cycle. So one area is life cycle management. When you come to industry, you will understand more about, people will be talking about, I'm in regulatory affairs, I'm working in life cycle management. Life cycle management is nothing. from development to the product obsoleteness, you have to maintain all the data. So that is one of the areas you people can work. Now, mandatorily, each company has to have its own pharmacovigilance department with a clinical pharmacist and a medical doctor, and you should have your own SOPs as well as PMS system. And then what we will be doing is, any new drug in India which is approved, they have to do PSUR data, periodic safety update reports. And when I am manufacturing from another manufacturer, I need to see that my product is safely manufactured. So I have to put a safety data exchangement agreement, so SDA. So pharmacovigilance role is very huge. So that is one of the career options you can take. Again, this one is mostly people prefer. This is the dossier area. So dossier area is very easy for people coming from pharma background, because you are compiling the data. You should have a little knowledge on quality assurance or quality control. Initially, you work in quality, then come to regulatory for this particular profession. So here it is like ROW countries have 4 parts. Regulated markets have 5 parts. So the 5th part difference is common. One is the general admin part. Part 2 will be quality, non-clinical and clinical. In regulated markets, you have quality overall summary. So that will have every overview completely written, and it will be mostly into ECTD format. Let me tell you, nowadays even a small license application from domestic is online. So when you have an online skills, definitely applications are very easy. So most of the things you have to verify the documents, and then reviewer will be there, publisher will be there, and then the document goes to the agency. US, they are very, very clever, because they don't want to see the files second time. So it will be in XML format. So each format will generate one particular code. So next time if the code comes in, they will see that you have changed that. So date, revision, everything will be there. So you are not supposed to touch once you have published the data. Before that you can do that. So I insist, if something regulatory has to happen, you can talk with a lot of dossier development teams. So they give you the software for less price, so that colleges can practice that. It will be very useful for you when you come to the job. And new drug in India, it's very much similar only. You have to get a clinical trial approval, and then e-filing. Now we had online Sugam, and then now that NSWS has come into. So based on the study data, the new drug applications will be there. Here we have a scientific expert committee meeting. You have to submit, drug application, we all know that license. What are the other licenses? You have subsequent new drug, you have fixed dose combination, you have new drugs. So there are a lot of subcategories. Have you ever visited CDSC website? Anybody? No. Just type in cdsco.gov.in and look into the different types of application. There are huge list of applications. See, 5 websites: CDSCO, FSSI, NAPA, and then whatever research websites you are browsing, apart from that FDA or EMA. Just open it and see that what are the new updations, what all the changes they have brought in. So it will be very interesting for you for your future also. Again, this is one of the area where people are placed. Packaging, labeling. So we've seen that lot of packaging labelings, in abroad it will be like SMPC. It is called as summary product characteristics. So you have to give all the pharmacology data, whatever has been generated for the product, and then you'll be also giving a pill, package insert. So that will be in the local language. So that will be very done by professional team. And labeling is also. So once labeling is not proper, what are all the cases the DA put for the product when they take a sample? One is, any guess? If label fails, it's misbranded. If quality fails, it is not standard quality, NSQ. And then if the product itself has admixture or the AP itself it's different, it will be spurious. So lot of regulations are there. So market complaint again is one of the area. So if you get some time, you can look into the Drugs and Cosmetics Act. So it will give you most of the information. So there I put one label. You can see the red line with RX means it's prescription drug. It has to be taken only by a prescription, with the prescription. So the labeling under Rule 96 will contain statement of net content, active ingredient, manufacturer detail, manufacturing license number, physicians, whether it is to be taken, caution, warning. So all these things will be in the drug list. So these are the new packaging informations. No, Schedule G was there, H1. In H and H1, they will give you list of ingredients which will fall under H and H1. What are all the prescriptions should be done? See, when we want to control AMRs, first of all, we have to implement these kind of activities, because these warnings will tell them that not to buy, not to sell without prescription. Certain drugs, only with prescriptions only they can dispense, the pharmacist can dispense. So this is one of the areas DCGI, they are working towards curbing all this resistance. We see a lot of logos when we go to the shop and pick that. You know, product contains vegetarian and non-veg logo. Have you thought about it? FSSAI products, most of the products will have a green dot as well as a brown dot. A green dot represents it's a vegetarian product, so it doesn't have any animal content or fish or something. So free from all this. So you can, next time you can see that, no, if a product is green dot, it's vegetarian. If it is a brown dot, it's a non-vegetarian. Of course, vegan has its own thing. Organic food is certified. AgMark is certified. Then you will have FSSAI number. Fortified is nothing but all the vitamins and complements. These minerals are in a coated kind of stuffs. Fortified would be different. This symbol will be there. Gluten again, no, that will be in this symbol. It's gluten free. Recently, we heard about this instance, no, the Ghana tragedy, no, cough syrups people have taken and the child have died. There were a lot of reports, no, concerning Indian quality systems and all. And of course, another was one, it was also from Chennai based company. They had some eye drops which caused, infections and blindness and one death. So because of this, the quality assurance part becomes very, very critical, and agencies will be often auditing the plants. So what is a CAPA? CAPA is a corrective and preventive action. So whenever you do an investigation, again, quality assurance investigation is one of the areas you can train yourself. I'll just show one incident. It is kind of, which come across in few companies. So there was a metal piece in a tablet. Once the inspection was done, what might be the possible reasons? Maybe, somebody must inspect, they have not done that. Or there was a braided rope in the FBD, kind of, this is a fluid bed dryer, so that might have fallen, that might have caused. Or metal detector, usually metal detector will be there. So when you, or maybe during the sifting somebody have hand rubbed that, it might have caused. The reason is like, it was a metallic piece from the braided rope in the FBD. So now that they have covered that, and probably they have changed so that there is no metallic features. Each and everything the agency will be looking. And similar example in water treatment plant, Pseudomonas in potable water. Water has been completely tested. It will be like, Pseudomonas, no, what is Pseudomonas going to do to us? Nothing. Maybe analyst error, he must have analyzed, but that might be an analyst error, or improper supervision. The supervisor have missed it. Or the chlorination is not sufficient. For Pseudomonas, chlorination will be done routinely. When you look into, or improper sampling, he has not taken enough sample to analyze. But the reason is, the chlorination is not sufficient because the resistance is growing with Pseudomonas. So we have to upgrade ourselves and change lot of agents to treat that. So now that companies have started using kind of cocktails of cleaning agents to make sure this is not occurring. And this is one of the interesting case. Efficacy in liquid dose. There was a market complaint. People said that earlier the efficacy was good, now the efficacy is not that up to that level. So you can say assay or uniformity failure, assay is good, uniformity is not, no issues. Or measuring up graduation is not proper. Yeah, that vendor is approved. He is an alternate vendor only. Not mixed well during consumption, so you have to shake well before use. Has he not shaken? No, he has shaken. And psychological thought of a patient, oh, I drink usually, but it's not working this time. That might be psychological. But the real reason when we analyzed was measuring cup. You know, the measuring cup graduations will be there. So that graduations will be like 1, 2, 3 ml, but sometimes that graduations will be little different because colored liquid, lower minuscule, normal liquid, this upper minuscule. So this kind of difference, that issue happened, so that the dose was taken less actually. So that was avoided. So you can see how many inputs quality assurance team can bring into this kind of studies. Of course, we have been talking about US have moved 100% ahead of us. So they have passed recently a Modernization Act 2.0 that they want to replace animal studies. So they want to bring in new technology, kind of in silico models, 2D culture, 3D, this organ on chip. They have already worked out and one particular model is also running. So henceforth, animal models will be reduced and they will be going for a non-animal invasive models so that this backlashes against usage of animals is prevented. So this is almost there, and one of the such tool is AA. So this is one of our product which we launched during COVID. So once the COVID, they have approved on the MPRO is the docking site. So we did a docking study. So once based on the docking study, then we identified that and we have done a clinical trial. So based on this drug is called as repurposed drug. So usually India new product is less, but repurposed drug will be there. COVID has taught us that already drugs are there. You can repurpose it to a different indication. So again, you can go to library, you can go for search, see the pathway, whether that pathway acts in 2 different areas. You can select that pathway and develop a drug for a newer repurpose indication. So that is what $1 million. So this drug was approved by the government for mild to moderate. We have done in Madras Government Hospital, 100 patients, and it was one of the drugs approved for a mild to moderate treatment. Again, this India Pharma compliance with FDA, there were a lot of cases in 23. It was around 117 cases voluntary action. So usually US will give you official action or voluntary action. There I will accept and go. But here, what will say? There, if I ran on a throne, I will say that I ran the throne. What here will say? Nammala mullu kuthirchu appadimo. That came and pinches, but they will have a voluntary recall system. They are fine with that. But in India, people won't accept that. But still, FDA is insisting on that. By 2024, the cases fell to 14 actually. From 117 cases to 14, that was a very big improvement from the side of Indian manufacturers. So we are ensuring high quality manufacturing practice as of now for the FDA, particularly for Indian inspections. Again, this regulatory affairs trend is changing towards a lot of automation, opportunities of harmonization, clinical trials. So I think you can consider this as a seriously a profession and take it up. Probably my last slide. So this kind of earlier it was like I need to all these quality parameters, government policies, governance, laws, standards, everything is to comply. I have to be regulatory wise, I should comply. But now it has changed. Industry, regulators and regulatory compliance are towards patient safety. So patient safety will be the next thing. All the industries and companies, regulators, the government will be focusing on. So with this, I thank all of you for a patient hearing. Thank you.
Thank you, sir. It was a wonderful session navigating through the role of regulatory profession in pharma industry, ICH guidelines, automation in regulatory affairs, etc. I hope it will help our upcoming graduates. Any queries, please? Any questions? Any questions from the audience? So, I have a few questions regarding So regarding the fast track approvals, whether it will compromise the patient safety, especially in pandemic conditions and all?
Sir, usually no, because statistical relevance for any clinical trial is very important. So we have a lot of methods, chi-square test. So there are a lot of statistical methods which give you the thing. Here, what I am talking about fast track during COVID and other things is like if the product is already established for quality and safety. So directly it is only a repurposing drug. So only you have to do a activity. For example, in COVID, hamster model. If you do in hamster model itself, government was willing to launch that product. So hydroxychloroquine they done in hamster model only. In US, for us, it's like rare disease. It's like in 100,000 people, there is 1 lakh people, only 10 people are affected. So nobody can find that much population. So it will be on a lesser population. So that way they take a statistical relevance.
Thank you, sir. And so the session is concluded. Thank you.
Thanks.
Here we end the session and we are taking a moment to present our speaker, Dr. L. Ramanathan, with a token of appreciation and our gratitude for such a detailed and informative presentation. For doing the honor, I invite the Principal of Holy Grace Academy of Engineering, Professor Dr. Prashant, onto the dais. Please, sir. Doing the honor is the Principal of Holy Grace Academy of Engineering, Professor Dr. Prashant. To honor our chairperson, Dr. Shyam Kumar, Principal, LSR College of Pharmacy, I invite Mr. K.T. Benny, Holy Grace Academy of Pharmacy, Secretary. Please, sir. Thank you, sir. With that, we end the forenoon session and we are dispersing for the lunch break. The lunch is arranged both at the first floor and the second floor. Delegates, please take the exit way. Our student volunteers will guide you to the lunch break hall. We are announcing the prizes for e-poster presentations. So before announcing the prizes of e-poster presentation, we are taking a moment to present a token of appreciation to the evaluator of e-poster presentation, Dr. Sapna Srikumar, Principal, Ahalya College of Pharmacy. For doing the honor, I invite Professor Dr. Jayashekhar sir, the President of Kerala State Branch IPA, for presenting her with the certificate of appreciation. Please on to the stage, sir. Doing the honor is Professor Dr. Jayashekhar, President IPA Kerala State Branch, to Dr. Sapna Srikumar, Principal Ahalya College of Pharmacy. Moving on to the prizes for e-poster presentation. For the third prize is backed by Aisha Mirfa of Holy Grace Academy of Pharmacy. I request the speaker for the next session, Mr. Manu M, to hand over the prize to Ms. Aisha Mirfa. And the second prize goes to Jittu Mohan of Sanjo College of Pharmaceutical Sciences for presenting the certificate and cash prize for the second prize of e-poster presentation. I invite Dr. Ajit Kumar P, the Academic Director, Holy Grace Academy of Pharmacy, onto the stage. Please, sir. And the first prize for the e-poster presentation competition is backed by Michelle Ashik of Karpagam College of Pharmacy, Coimbatore. I invite Professor Dr. Jayashekhar to hand over the certificate and cash prize to the first prize winner. Please, sir. I invite Professor Dr. Jayashekhar to hand over the certificate and cash prize to the first prize winner, Mr. Michelle Ashik of Karpagam College of Pharmacy, Coimbatore. Congratulations to the winners of e-poster presentations. Now we are moving on to the next session. Before that, I request all the delegates to occupy the seats at the front of the stage. Please move forward to the front seats of the hall.
So now we are moving on to the afternoon session. For the first session, we have Mr. Manu M, Pharmacovigilance Service Specialist, Accenture Pharma India Private Limited, Chennai. And this session will be chaired by Dr. Anup KR, Principal, Triveni College of Pharmacy. I request the dignitaries to occupy the seat on the dais. I request the dignitaries to chair the session. Really. Sir, please have your seat. Dr. Anup KR, a distinguished academician and administrator with over 23 years of experience in the field of pharmacy education. He currently serves as a principal of Triveni College of Pharmacy, where he has been providing visionary leadership since 2015. Dr. Anup's career highlights include the tenure as Assistant Controller of Examination at Amrita Vishwa Vidyapeetham, where he was instrumental in academic and administrative reforms. Manu sir is from Kayangulam, Alappuzha, completed B.Pharm in 2014 from Academy of Pharmaceutical Science, Periyaram, Kannur. now known as Government Pharmacy College, Kannur Medical College. Sir has graduated his Masters in Pharmaceutics from the same college in the year of 2016. Manu M. Sir has started his career as clinical pharmacist in Apollo Cancer Institute, Chennai from 2016 October to 2018 of May. Later, Sir joined as Accenture Solutions as a drug safety associate in ICSR processing and in 2022 promoted as a team leader. Later, at present, he is currently working as pharmacovigilance service specialist, leading a team of 15 members at this institution. As we are honored to have Mr. Manu M as a resource person, so with a big round of applause, let's welcome Sir to this session. Over to you, sir.
Hi all, very good afternoon. Am I audible? Can you hear me clearly? Okay. So good afternoon once again all. Afternoon session, post lunch, all are feeling sleepy, right? Want that mood, right, everyone? Okay, fine. So first of all, as I said, myself Manu and I am from Accenture and I am working in the pharmacovigilance department for the past 6 to 7 years. So today we are about to discuss on some of the emerging trends or on the basics on pharmacovigilance. So I would prefer to continue in a mix-up of language. I'm okay to continue in English, but let's go for a mix-up of language that will be easy for you people as well. ഒരുപാട് ബോറടിക്കാതെ കാര്യങ്ങൾ നമുക്ക് മുന്നോട്ട് കൊണ്ടുപോകാമെന്ന് വിചാരിക്കുന്നു. ബോറടിപ്പിക്കാതിരിക്കാൻ പരമാവധി ശ്രമിക്കാം, ഉറക്കാതിരിക്കാനും നോക്കാം. ഓക്കെ. So first point, again understanding of PV and emerging trends, and this is just a disclosure where the content or the thoughts whatever we are going to discuss over here is the sole discussion for me and it does not represent sorry for that. So whatever the content or the thoughts we are going to discuss over here, this is a sole discussion for me, even though I belongs to Accenture, that does not represent any content or any thoughts which is part of Accenture. Okay, this is just a disclosure from my end. And if you are going to the topic, so the first one, before going deep into pharmacovigilance, maybe you people might have already gone through this drug development. So you know right what you mean by drug development or where we are starting drug development. So as usual in our laboratories or from a natural sources, that may be either from a plant or a biological source or an animal source, we will be identifying some chemical constituent or active ingredients. So once that got identified, we will be going for an animal study to prove the efficacy and safety of that particular drug. നമ്മുടെ സ്ഥിരം ബലിയാടുകളായിട്ടുള്ള ആൽബിനോ റാറ്റ്സ്, ആൽബിനോ മൈസ്, ഇവരൊക്കെ തന്നെ വീണ്ടും ഇവിടെയും വരും. ആൻഡ് വി വിൽ ബി ടെസ്റ്റിംഗ് അവർ ഡ്രഗ് ഓവർ ദെയർ. വൺസ് all the tests are completed, we will be going for another option, that is IND. Do anyone know what is IND? Come on, maybe some of you know that. Super, good. So it's investigational new drug. Again, what is investigational new drug? Okay, so whenever you are identifying something new, we will be, or we have to finalize, or we have to file a new drug application, that is IND, investigational new drug application. Along with that, this is some sort of a license where we can transport the active ingredient from one place to another. So whatever we are discussing over here is purely FDA norms. Each country will be having different norms based on their regulatory rules. Okay. So നമ്മൾ ഒരിക്കൽ IND ഫയൽ ചെയ്താൽ within 30 days we will be waiting for FDA approval until unless FDA is stating that is as a no. FDA no എന്ന് പറഞ്ഞില്ലെങ്കിൽ definitely we can go for next level of our drug development, that is clinical trials. Okay, so clinical trials you people might have already learned. നിങ്ങൾക്ക് എല്ലാവർക്കും അറിയാം what are the phases of clinical trial, what are the types of clinical trial, right? Phase 1, phase 2, phase 3, all those aspects. We will be having different volunteers, patients, control group, test group, all those aspects, right? So once it is coming to clinical trials, we will be checking on the efficacy of that particular active ingredient or that particular formulation, whatever we have identified in human, and we will be proving the safety and efficacy of this particular drug over here, right? So once the clinical trials are completed and we are completely safe with the efficacy of the drug, നമുക്ക് ബോധ്യമായാൽ after the clinical trial എല്ലാം basic ആണെങ്കിൽ, we will be going for NDA. What is NDA? Same, it's a new drug application. So once new drug application is a set of a big document that will be contained more than 10,000 pages. Okay, it will be a 10,000 page plus document. നമ്മൾ ഇത്രയും നാളും ചെയ്ത ഫ്രം ദ സ്റ്റാർട്ട് ഓഫ് ദ പ്രോസസ്, ഒരു ആക്റ്റീവ് ഇൻഗ്രീഡിയൻറ്റ് കണ്ടുപിടിച്ച് അതിൻ്റെ സ്റ്റഡീസ് നടത്തി അതിൻ്റെ പ്രൂഫ് കണ്ടുപിടിച്ച് വാട്ട് ആർ ഓൾ ഇൻഫർമേഷൻ വി ഹാവ് ഗാതേർഡ് ടിൽ നൗ will be coming in NDA and that will be filed for the approval. Okay, so once the NDA approval is completed, our product will be ready to be in the market, and the product will be going to the market after all the approval. Once the product is in the market, we will be getting a patent, right? For our actual product, ഒരു patent issue ചെയ്യും. And what would be the validity of the patent? 20 years. Okay, so 20 years would be the patent period. Till that time, we are the sole owners for that particular product. But after the patency period, again, the product will be going for an open market. ഇത്രയും കാലം ഞാൻ A+B=C എന്നുണ്ടാക്കി വെച്ചൊരു പ്രൊഡക്റ്റിനെ patency period expire ആയാൽ anyone in the market can take it up and do it like P+Q=C. Right? So that would be the expiry, that would be going to happen after the expiry of the product. So once the patent got expired, it will be going for open market. Once it is going to the open market, anyone in the market can take it up, and that will be filing as an ANDA, that is an abbreviated new drug application. So once ANDA also completed, anyone can manufacture it, anyone can bring it to the market. So again the product will be the market. So this is just a basic slide I usually kept in my slides to make you people understand about what is the drug development part alone. Okay, so maybe you already know some of these points, but I just want to highlight these things. Fine. So coming to pharmacovigilance, that is the basic process or basic thing we have to discuss over here. So the pharmacovigilance, I think it is a part of your syllabus now, I'm not sure. But previously, ഞങ്ങളെല്ലാം ബി.ഫാം പഠിക്കുമ്പോൾ ഫാർമക്കോളജി ടെക്സ്റ്റിൽ ഏതോ ഒരു പോർഷനിൽ STCത്തിലൊക്കെ ചെറിയൊരു പോർഷനിൽ മാത്രം കണ്ടിട്ടുള്ള ഒരു ഭാഗം മാത്രമായിരുന്നു ഫാർമക്കോവിജിലൻസ്. ഓക്കെ, സോ ഞങ്ങളെ സംബന്ധിച്ച് ഇറ്റ് ഇസ് എ 5 mark question, and for that we have to by heart this particular definition. So pharmacovigilance is the science of collecting, monitoring, researching, analyzing or assessing and evaluating the reports or the information whatever we are receiving from the healthcare professionals or a consumer or a patient, whatever it may be, regarding the adverse effects of a medication. This is the basic definition, and you may by heart it for your academic purpose and maybe for the interview purpose and all. So this is the basic definition of pharmacovigilance. If you are going deep into the terminology part and all, so pharmacovigilance is derived from 2 words. One is pharmakon, that is a Greek word which mentions like a drug. Pharmakon means like a drug, and vigilare, it is a Latin word which is mentioning like a keep watch. So if you are combining together, it will be something like keep watching of a drug. That is the basic we are doing in pharmacovigilance. So it's a science of collecting, monitoring, assessing, evaluating the safety and efficacy, and thereby preventing any adverse events or adverse effects of a particular medication in our market. This is the basic definition of pharmacovigilance. Okay. So, ഈ പിക്ചർ നിങ്ങൾ എല്ലാവരും കണ്ടിട്ടുണ്ടാവും. Pharmacology students or being final year B.Pharm students, most of you might have seen this picture, right? Can anyone identify what is this? Sorry? I couldn't hear you. Good. So it's phocomelia. Okay, thanks for that answer. So this particular condition is called as phocomelia. ഇതൊരു പക്ഷേ നമുക്ക് മനസ്സിലാകുന്ന മറ്റൊരു പേര് താലിഡോമൈഡ് ട്രാജഡി എന്നായിരിക്കും. യു മൈറ്റ് ഹാവ് ഹേർഡ്, റൈറ്റ്? താലിഡോമൈഡ് ട്രാജഡി. വാട്ട് ഈസ് താലിഡോമൈഡ് ട്രാജഡി? എന്താണ് താലിഡോമൈഡ് ട്രാജഡി? വൈ ഇറ്റ് വാസ് ഹാപ്പൻ, ഹൗ ഇറ്റ് വാസ് ഹാപ്പൻ, എങ്ങനെയാണ് ഇത് സംഭവിച്ചത്? എനി ഐഡിയ? Okay, fine. So this picture you have understood, right? Most of you might have during your B.Pharm or during your medicinal life, you might have already seen this particular picture. So this condition is called as phocomelia. Phocomelia is caused by thalidomide, a drug which was used for the indication of morning sickness in pregnant ladies during 1950s. Okay, 1950s pregnant ladies use ചെയ്തുകൊണ്ടിരുന്ന ഒരു ഡ്രഗായിരുന്നു താലിഡോമൈഡ്, യൂസ്ഡ് ഫോർ ദ മോർണിംഗ് സിക്നസ്. ബട്ട് അൺഫോർച്ചുനേറ്റ്ലി ആ സമയത്ത് whomever taken thalidomide drug, the children or the fetus coming out of the pregnant ladies had this particular condition as phocomelia, which is with sealed limbs or absent limbs. Okay, as you can see the picture, പേഷ്യൻ്റ് അല്ലെങ്കിൽ ബേബിയുടെ ലിംസിൻ്റെ ഷോർട്ടൻഡ് ലിംസ് അല്ലെങ്കിൽ സംടൈംസ് അവിടെ ഒരു പോർഷൻ ഇല്ലാതെ തന്നെ വന്ന കണ്ടീഷൻസ് ഉണ്ടായിരുന്നു. സോ ദാറ്റ് ദേ ഹാവ് ബാൻഡ് താലിഡോമൈഡ് ഇൻ 1960s because of this particular issue. Okay, so I'll park this particular picture over here. We will come back to this, but still let's move on. Now, why pharmacovigilance? Okay, so why we need to perform a pharmacovigilance? So before going deep into that, I just want you people to have some imagination. Basically, whenever we are saying something, think on the way we are taking a drug that is getting administered to the patient, and we are assessing the efficacy and safety of that particular drug in that patient. ഒരു പേഷ്യൻറ്റിന് ഏതോ ഒരു ഡ്രഗ് കൊടുക്കുന്നു, ആ ഡ്രഗിന് ആഫ്റ്റർ എന്ത് സംഭവിക്കുന്നു എന്നതിനെ പറ്റിയുള്ള ഒരു അസസ്മെൻറ്റ് ദാറ്റ് വി ആർ ഡൂയിങ് ഓവർ ഹിയർ. ഓക്കെ, so this is one basic instance or one basic reason we have initiated pharmacovigilance in all the areas. ഇങ്ങനൊരു ട്രാജഡി സംഭവിച്ചത് the big reason, right? ഇപ്പോഴും there are remainings for this happening for that particular drug. Okay, so why pharmacovigilance in pre-marketing safety data? So as we discussed earlier, we are doing something like an animal studies, right? So how much effective or how much you can utilize or depend on animal studies? എത്രത്തോളം we can able to extrapolate the information whatever we received from animal studies. നമ്മൾ നേരത്തെ പറഞ്ഞ പോലെ എലിയിലും അല്ലെങ്കിൽ ഗിനി പിഗ് ഓർ റാബിറ്റ്സ് ഇതിലൊക്കെയാണ് നമ്മൾ ടെസ്റ്റ് ചെയ്യുന്നത്, റൈറ്റ്? ബട്ട് ഒരു ഡ്രഗ് ടു ഹൂം വി ആർ ഗോയിങ് ടു ഗിവ്, നമ്മൾ കൊടുക്കാൻ പോകുന്നത് പേഷ്യൻസിനാണ്, ഹ്യൂമൻസിനാണ്. സോ ഹൗ മച്ച് വി ക്യാൻ എക്സ്ട്രാപ്പുലേറ്റ് ദിസ് പർട്ടിക്കുലർ ഇൻഫർമേഷൻ ടു ഹ്യൂമൻസ്? ഒബ്വിയസ്ലി ദർ വിൽ ബി ഡിഫറൻസ്, റൈറ്റ്? സ്വാഭാവികമായ എന്തായാലും ഡിഫറൻസ് ഉണ്ടാവില്ല. ഒരു അനിമലിൽ ഉണ്ടാവുന്ന ഒരു ആക്ഷൻ ആയിരിക്കില്ല. ഒബ്വിയസ്ലി ദാറ്റ് വിൽ കം ടു ദ ഹ്യൂമൻ പേഷ്യൻറ്റ്. സോ we can't completely say like animal experiments are relevant in pharmacovigilance or completely can be utilized in pharmacovigilance. Okay, so that is the first point. And second point, whether all the clinical trials, so we are doing clinical trials to avoid all such information, but whether all these clinical trials are complete, we would be able to get all the information from clinical trials. Can we get that? Okay, fine. Let me ask you one question. You might have already heard about concordant values, right? The concordant values. In the chemistry labs, most of the time you might have used titration chamber, right? So how many of you really did the concordant value and get it submitted to your lecturer and get it signed off? Concordant value kittunnam vare titrate ചെയ്ത് ആ വാല്യൂ കൃത്യമായി കിട്ടിയതിന് ശേഷം മാത്രം പോയി സൈൻ മേടിച്ച് എത്ര പേരുണ്ടാവും? ഫൈൻ. ഞങ്ങളൊക്കെ പഠിക്കുമ്പോഴും അങ്ങനെ തന്നെ ആയിരുന്നു. ഓക്കെ, ഏതർ നമുക്ക് നേരത്തെ ലാബ് കഴിഞ്ഞ് വീട്ടിൽ പോകണം, അല്ലെങ്കിൽ ചിലപ്പോൾ ടീച്ചർ നേരത്തെ പോകണമായിരിക്കും. വൺ ഓർ ദ അതർ വേ കൺകോർഡന്റ് വാല്യൂസ് എല്ലാം നമ്മളിങ്ങനെ കണ്ടുപിടിച്ചുകൊണ്ടിരിക്കുകയായിരുന്നു. ഒന്ന് കിട്ടിയാൽ തന്നെ സന്തോഷം, ബാക്കിയുള്ളത് നമ്മൾ തന്നെ തന്നെ ഉണ്ടാക്കിക്കോളാം, റൈറ്റ്? സോ ദാറ്റ് വാസ് ദ അപ്രോച്ച് വി വെർ ഫോളോയിങ് ലോങ് ബാക്ക്. ബട്ട് ഇതേപോലെയുള്ള നമ്മളിൽ ചിലരാണ്, ഫോർ സം ഓഫ് us വിൽ ബി ഗോയിങ് ടു ക്ലിനിക്കൽ റിസർച്ച് ആൻഡ് ഡൂയിങ് ദിസ് ആക്ടിവിറ്റി, റൈറ്റ്? ഐ എം നോട്ട് സേയിങ് ഇറ്റ് ആസ് എ മാനിപ്പുലേഷൻ, ബട്ട് സ്റ്റിൽ അവിടെ നമ്മൾ ചെയ്യുമ്പോൾ വി വിൽ ബി മോർ കോഷ്യസ്, മോർ വിജിലൻറ്റ് ഡൂയിങ് ദാറ്റ്. ബട്ട് സ്റ്റിൽ there are possibilities for human error that can happen, right? So we can't completely depend on clinical trials that as a complete data. And moreover, as you learned, we will be doing the studies in max out 500 to 1000 people, or depends on the number of population whatever we are having. അത്രയല്ല നമ്മുടെ പോപ്പുലേഷൻ, അല്ലെങ്കിൽ നമ്മുടെ ഡ്രഗ് റിസീവ് ചെയ്യേണ്ട പോപ്പുലേഷൻ അത്രയല്ല. ഇറ്റ് വിൽ ബി ഗോയിങ് ടു എ ലാർജർ പോപ്പുലേഷൻ. സോ ഹൗ ദാറ്റ് എഫക്ട് ക്യാൻ ബി കംപ്ലീറ്റ്ലി അനലൈസ്ഡ് ഓവർ ദി. So we can't completely say like all the clinical trials will be giving complete information regarding a particular drug. Okay, so that is the basic reason on the pre-marketing data. Now, why pharmacovigilance in post-marketing topics? So I have listed few things over there. So first one, unexpected adverse reactions. So before going to that, maybe some of you might have gone through pharmacy, dispensing areas on road, or at least some medicines you might have handled. You might have seen some leaflets or package inserts. കണ്ടിട്ടുണ്ടാവും, വായിച്ചിട്ടുണ്ടാവും, റൈറ്റ്? കണ്ടിട്ടുണ്ടോ ലീഫ്ലെറ്റ്സ്, പ്രോഡക്റ്റ് ഇൻസേർട്സ് എന്ന് പറയുന്ന സാധനം? സോ ഇറ്റ് ഈസ് അവൈലബിൾ ആസ് എ ഒരു ചെറിയ കഷ്ണം പേപ്പർ. എല്ലാ പ്രൊഡക്റ്റിൻ്റെ കൂടെയും അല്ലെങ്കിൽ ഏതോ ഒരു ബോക്സിൽ, ഏത് മാനുഫാക്ചറിങ് ചെയ്താലും ആ ഒരു ബോക്സിൽ ഒരു ലീഫ്ലെറ്റും ഉണ്ടാവും. ദാറ്റ് വിൽ ബി ഹാവിങ് ദ കംപ്ലീറ്റ് ഇൻഫർമേഷൻ റിഗാർഡിങ് ഓർ ദ പ്രിസൈസ് ഇൻഫർമേഷൻ റിഗാർഡിങ് ദാറ്റ് പർട്ടിക്കുലർ ഡ്രഗ്, അതിൻ്റെ ഇൻഡിക്കേഷൻ, അഡ്വേഴ്സ് ഇഫക്ട്സ് ഓർ ഡോസ്, റൂട്ട്, ഓൾ ദോസ് ആസ്പെക്ട്, ഇതെല്ലാം ആ പാക്കേജ് ഇൻസേർട്ടിലുണ്ടാവും. സോ ഈവൻ ദോ ഈ പാക്കേജ് ഇൻസേർട്ട് വി ആർ ഡിഫൈനിങ് ഓർ വി ആർ ഡിറൈവിങ് based on the clinical experiments or whatever the trials we have performed, and we are keeping it as a drafted. But still, ഇതെല്ലാം ഉണ്ടെങ്കിലും there will be possibilities for unexpected adverse reaction that can happen to a patient. നമ്മൾ നേരത്തെ പറഞ്ഞതുപോലെ the clinical trials are doing in a number of population, 500 to 1000 people or 1000 to 2000 people, right? ഇതല്ലാതെ we may be having multiple patients involved and multiple unexpected reactions also happening to a patient, right? There is a possibility, no? So that's why we need pharmacovigilance in post-marketing data. Okay, and coming to the next topic, that is interactions. This will be familiar for you, right? What do you mean by interactions, or what are the types of interactions? drug-drug interactions, drug-food interactions, right? So if you are taking examples for food interactions, so common example, most of you learn like tetracycline should not be given with dairy products, right? You might have heard or you might have learned. So obviously the dairy products may be reducing the absorption of tetracycline, that may be going to an inefficacy of the drug. Again, grape juice. So most of the drugs should not be consumed along with grape juice, that will be affecting the absorption or pharmacokinetics of that particular drug. These are some examples of food drug interactions. So we need to study on that. Again, coming to drug-drug interactions. In your pharmacology, you might have learned about synergism, antagonism, all those aspects, right? What does it mean? What is the significance of that? A drug A can increase the efficacy of B or can suppress the efficacy of B. Definitely that also can happen, right? Some drugs may be having a potential efficacy, but if it is given with another drug, that particular efficacy can be reduced for that particular patient. There is possibilities. So we need to learn about that also. So drug-drug interactions or drug-food interactions, or what are all the interactions available for us, we need to learn from that. And coming to the next thing, risk factors. So what are the basic risk factors whenever you are administering a drug to a patient? Again, you can extrapolate the clinical data information. You are doing only for 1000 patients. But this particular drug have to be given to pediatric patients, elderly patients, or the patients with hepatic impairment or the renal dysfunctions. All can be given with same dose? No, right? There should be some modifications. Otherwise, that will be harmful to the patient rather than an intended benefit. Correct? We are giving for some particular benefit, but the patient is having an hepatic dysfunction. Definitely that drug will not be metabolizing in our body, that will be leading to a toxic effect. Right? So that have to be studied. So that is the one point on risk factors. Similarly, age group or the disease condition of the patient, all those aspects. Okay. So coming to the quality of life, how a drug will be affecting the quality of life. So this is basically for a lifestyle medications or lifetime medications. So maybe hypertension, diabetes mellitus, all these are lifelong disease conditions, right? Are we getting any cure on that once hypertension is diagnosed? Lifelong continue, right? So I'm just taking an example of amlodipine. So if a patient is using amlodipine for a longer period, the studies shown like that patients or that humans will be having dry cough as a consequences for that particular effect on a long term effect. So definitely that will be going to affect the quality of life of a patient or a human, right? Similarly, multiple drugs also. So that's why we need pharmacovigilance in quality of life. Now coming to the next point, long term efficacy. Again I am saying again a lifestyle disease. Say for diabetes mellitus, what would be the primary drug of choice for diabetes mellitus? Metformin, right? So you are giving metformin as a primary disease or primary therapy for a patient. You are giving for 1 year, 1.5 years. After 1.5 years, metformin 500 may not be sufficient to control the blood sugar of the patient, right? So there will come the primary failure of that particular drug. You may have to add glimepiride or glipizide, something like that. Again, that will also go for 1 year, 1.5 years. Again, it will be dropping to the or not sufficiently controlling the blood sugar. Again, secondary failure. You have to prefer insulin. So the long term efficacy definitely will be questioned, and there will be primary failures or secondary failures of therapy regimen, especially in sort of things. And again in oncology conditions as well. So cancer treatment, whenever the patient is taking, definitely there will be limitations to get complete response for a cancer patient from a particular regimen. So we may have to switch the regimen, change the regimen, add on drugs like that. Okay. And to the last point, that is cost assessment. Very simple. For a patented product, definitely will be having more cost. Right? നേരത്തെ നമ്മൾ പറഞ്ഞ drug development until unless it is open to the market, അല്ലെങ്കിൽ ഇനി ഒരാൾ അത് A+P പകരം P+Q ഉണ്ടാക്കാന്ന് വരെ, there will be a much cost for that. അത് എല്ലാവർക്കും affordable ആയിരിക്കണം എന്നില്ല. So the cost assessment part also need to be assessed in pharmacovigilance. Okay? So Okay, so this is just some articles, whatever I have referred. I am just leaving the slide as such. Now coming to the advancement of pharmacovigilance, or why we need on the extrapolation of pharmacovigilance. First point is the risk-benefit analysis. What do you mean by risk-benefit analysis? Go ahead. You might have learned it, risk-benefit analysis in your college classes and all. Nothing, very simple. Whenever you are giving a drug, the benefit should be always high rather than a risk. Then only you have to give that, right? നമുക്ക് വേണ്ടാത്ത അല്ലെങ്കിൽ നമുക്ക് ബെനിഫിറ്റ് തരാത്ത ഒരു സാധനവും നമ്മൾ കഴിക്കേണ്ട ആവശ്യമില്ല. അറ്റ്ലീസ്റ്റ് റിസ്കിനേക്കാൾ കൂടുതൽ ബെനിഫിറ്റ് തരാത്ത ഒരു സാധനം നമ്മൾ കഴിക്കേണ്ട ആവശ്യമില്ല. സോ ഓൾവേസ് ദ റിസ്ക് ബെനിഫിറ്റ് റേഷ്യോ ഷുഡ് ബി 1 or less than 1. Then only we will be preferring that particular drug for this particular patient. Otherwise we will not be. So the risk benefit analysis is the basic thing. It should be always 1 or less than 1. That should give benefit to the patient rather than a risk. Clear? Aligned? Okay. So now the next one is on the rebirth of drugs. What do you mean by rebirth of drugs? Now I am going back to my older slide. Phocomelia. Why phocomelia caused, or why thalidomide caused phocomelia? What was the action happened over there? Any guesses or any idea? What had happened when patient taken thalidomide? What had happened to cause this particular issue? Okay, so thalidomide did one action, that is inhibition of angiogenesis. Okay, so inhibition of angiogenesis. Angiogenesis is nothing but, angio means related to blood vessels, genesis means the formation of blood vessels. So what thalidomide did in these patients? They have inhibited the formation of blood vessels in the baby, which lead to phocomelia or sealed limbs or absent of limbs, all those aspects. Now coming to the vast topic, why we need a rebirth of drug. So pharmacovigilance associate that time, they think about thalidomide can do this. They can inhibit the angiogenesis. So why can't we utilize this on another way? So they have utilized that. How can we use it? The inhibition of angiogenesis, how we can use in another way? Any guesses? Okay, so inhibition of angiogenesis we can use in cancer patients for the targeted cell therapy. Okay, so nowadays this particular thalidomide, whatever banned from 1960, now it is efficiently used in multiple myeloma conditions in most of the patients, along with its derivatives, thalidomide, lenalidomide, pomalidomide, whatever being available in the market, we have derived it. Based on that particular side effect, we are utilizing that in multiple myeloma for a larger population. So that is called by rebirth of drugs. Or otherwise again, along with that you can utilize, how we can utilize the side effect of a drug to a better option. You might have heard about minoxidil, right? Right? What is the basic action of minoxidil? It's a antihypertensive drug. Right? It's a classification by antihypertensive drug. But nowadays, minoxidil is most commonly used in hair loss treatment. Maybe you know that, right? It will be available with spray bottle or droplets or something like that. Minoxidil is causing a side effect of increased hair growth. So we have utilized it on that way. അതിനെ നമ്മൾ അങ്ങനെ യൂട്ടിലൈസ് ചെയ്തു. ആൻറ്റിഹൈപ്പർടെൻസീവിന് പകരം we have utilized it as a hair growth improver. That can also be done both. Okay, and now to the legal requirements. So definitely each country, so what is the regulatory authority for India? CDSCO. So each regulatory authority or each country will be having their own regulatory authorities, their own regulatory norms. So FDA, EMA, CDSCO, all those countries will be having. So different sort of regulatory authorities. Okay, China is having PMD as their regulatory authority, US is having US FDA, European Union is commonly having EMA, European Medicine Agency as a health authority. So everyone will derive their own regulations, their own rules for this particular drug, safety, efficacy, everything. Okay, so legal requirements obviously will be there for that. Now coming to basic understanding of PV. ഇതുവരെ ബോറടിച്ചോ? ഇല്ല, ചിലപ്പോൾ ഇച്ചിരി ബോറടിക്കും. ഓക്കെ, കാരണം these are completely going for the terminology related things. So how you will be analyzing or understanding the pharmacovigilance things? First thing, simple question, what is the difference between side effects and adverse effects? Side effects and adverse effects. എല്ലാ ഡ്രഗിനും സൈഡ് എഫക്ട്സ് ഉണ്ടാവും, ദാറ്റ് യു നോ റൈറ്റ്. ഐ മീൻ എല്ലാ ഡ്രഗും ഇല്ല, മോസ്റ്റ് ഓഫ് ദ ഡ്രഗ്സ്, റൈറ്റ്? All the side effects may not be adverse effects, but all the adverse effects will be a side effect. So side effects can be positive or negative. So negative ആയിട്ടുള്ള സൈഡ് എഫക്ട്സിനെ മാത്രമേ നമ്മൾ അഡ്വേഴ്സ് എഫക്ട്സ് അല്ലെങ്കിൽ അഡ്വേഴ്സ് ഇവൻറ്റ്സ് കൺസിഡർ ചെയ്യൂ. Others will be remain as side effects. Okay, so that is the basic thing. And again, so ഞാൻ നേരത്തെ പറഞ്ഞല്ലോ, you have to visualize something on, you are giving a drug to a patient, the patient is having some experience or experiencing some adverse events. Now we are categorizing these adverse events based on their seriousness. You can classify it as serious or non-serious. So you are administering a drug, a patient experiences an event. Now you have to analyze how much it got impacted to the patient, how much it got affected to the patient, whether that will be resulting in death of the patient. Then that is a seriousness criteria for the patient. Or it is going to a life-threatening condition. The patient may need to admit to an ICU or ventilator support or oxygen support, something like that. Coming to hospitalization, whether that particular adverse event caused the patient to be in hospital or prolonged hospitalization, we have to think on that way. And the next one, result in persistent significant disability, maybe some amputation or some sort of things, whether happening or not. Last one or the fourth one, result in congenital anomalies. You might have already seen thalidomide, whether it is causing that particular effect or not. And to the last one, there will be some adverse events that may not be fitting into this seriousness criteria, but will be considered as serious. ഒരു പേഷ്യൻ്റ് അഡ്വേഴ്സ് ഇവൻ്റ് ഉണ്ടായി, ദ പേഷ്യൻ്റ് മേ റിക്വയർ ആൻ ഇൻട്രാവീനസ് ഇൻഫ്യൂഷൻ ആസ് എ ട്രീറ്റ്മെൻറ്റ്. Definitely that is a serious condition, right? So based on that, we will be assessing that as other important conditions as well. So these are the criteria we are fitting into seriousness of an event. Okay? So now other events, whatever the rest of the events. I am just taking an example. You are getting, for a drug എടുക്കുന്നു, you are getting a rash or something like a nausea or vomiting. So you can think on the severity of that particular event, how much it will be severe. But there will be possibilities the patient may have or may develop ulcerative colitis or something like that, that may require a hospitalization. Based on that, we have to derive this, or we have to fit into this particular criteria. Okay, so this is again understanding of PV, unlisted and listed. So listed things already you are having in your package insert. ഞാൻ നേരത്തെ പറഞ്ഞില്ലേ, we are all having a product insert in our products. അതിൽ തന്നെ ഉണ്ടാവും what are the adverse event we can expect from this product. When you are taking this product, definitely you will be having so and so. നിങ്ങൾ ചിലപ്പോൾ കണ്ടിട്ടുണ്ടാവും. Maybe in your college you might have learned that. Same way. So there will be some unexpected events as well, which is not even listed. ഞാൻ ഒരു 1000 പേഷ്യൻസ് ക്ലിനിക്കൽ ട്രയൽ ചെയ്തു, but I could not able to identify that particular effect. Definitely that can happen. എനിക്ക് സംഭവിക്കുന്ന പോലെ ആയിരിക്കില്ല ഇനി ഒരാൾക്ക് സംഭവിക്കുക. So that is coming under unlisted or unexpected things. And the listed things, again the listed things will be coming as whatever already there in the product insert, that we know. എൻ്റെ ഡ്രഗ് എടുക്കുമ്പോൾ ഇന്നെല്ലാം സൈഡ് എഫക്റ്റ് വരും എന്ന് എനിക്ക് ഓൾറെഡി അറിയാം. That my clinical trials have proven. Fine. So this is the understanding on listedness and listedness and unlistedness. Now again, this is again the same thing, challenge, de-challenge and re-challenge. So challenge is nothing but if you are giving a drug to a patient, that is a challenge. Just the terminologies I am going, okay, nothing big on that, but you can understand on that way. Whenever you are giving a drug to a patient, that is a challenge. So administration of a suspect, that is a product by any route. We are challenging a patient by this particular drug. Now this come to the de-challenge. So once the patient taken the medication, there will be some adverse events. I am just taking an example. There will be some adverse events. So now you are going for the de-challenge part. De-challenge is nothing but you have to stop that drug. Challenged. Now you have to de-challenge it. So it will be coming as two classification, whether it is positive or negative. നമ്മുടെ ഡ്രഗ് കാരണമാണ് ആ എഫക്റ്റ് ഉണ്ടായതെങ്കിൽ, ആ അഡ്വേഴ്സ് ഇവൻ്റ് ഉണ്ടായതെങ്കിൽ ഡെഫിനറ്റ്ലി നമ്മൾ ആ ഡ്രഗ് സ്റ്റോപ്പ് ചെയ്യുമ്പോൾ ആ ഇവൻറ്റും സ്റ്റോപ്പ് ആയിരിക്കണം. റൈറ്റ്? ദാറ്റ് വിൽ കം ആസ് എ പോസിറ്റീവ് ഡീചാർജ്. വൈസ് വേഴ്സ, നമ്മുടെ ഡ്രഗ് സ്റ്റോപ്പ് ചെയ്തിട്ടും ദ ഈവൻറ്റ് ഈസ് കണ്ടിന്യൂയിങ്, ദർ ഇസ് നോ ഡിഫറൻസസ് ഓർ ചേഞ്ചസ് ഹാപ്പനിങ് ടു ദാറ്റ് ഈവൻറ്റ്, then it will be a negative rechallenge. Okay. So again rechallenge. So we given the drug, challenge happened, rechallenge happened. Now we have to verify it again. So we are rechallenging it. We are reintroducing the same drug on the same way to the same patient, and we will be analyzing. Now that is also for two things: negative rechallenge or positive rechallenge. If we are getting the same effect, then it's a positive rechallenge. If we are getting different things, then that is a negative. Obviously that particular drug will go for a reconfirmation or reapproval. Why? Because already ഒരു എഫക്റ്റ് ഉണ്ടായെന്ന് നമ്മൾ പ്രൂവ് ചെയ്തു, വീണ്ടും നമ്മൾ ഇൻട്രൊഡ്യൂസ് ചെയ്യുമ്പോൾ വി ആർ നോട്ട് ഗെറ്റിംഗ് ദാറ്റ് എഫക്റ്റ്, വിച്ച് മീൻസ് ദാറ്റ് ഡ്രഗ് ഈസ് നോട്ട് അപ് ടു ദ മാർക്ക്. ഓക്കെ, സോ ദാറ്റ് ഈസ് ഓൺ ദ ചലഞ്ച് ആൻഡ് റീചലഞ്ച് പാർട്ട്. നൗ the basic areas of PV, that is two different areas, that is ICSR and PBRR or PEBRR, okay, or PSUR. So the ICSR is the area what I am working for the last 5 years. So it is nothing but individual case safety reports. Okay, now I will explain you on later on that. So individual case safety report is nothing but if a physician or a pharmacist is reporting something regarding a drug to me, I have to develop that one individual case. That particular receipt or that particular thing will be called as a case, individual case, and we have to report this particular individual case to the regulatory authorities. Okay, I'm just saying the outlier of this. So whomever, a physician is reporting an adverse event regarding a drug for my product, and I am creating a case or I am preparing a case or a data sheet, and I am sending it to the regulatory authorities. That is ICSR. That's the area I am focusing on or I am working in. Now coming to the second part, that is PBRR or PSUR. Whatever I have sent to the regulatory authorities, they will be having a periodic analysis of these reports. ഞാൻ ഒരു മാസം 100 റിപ്പോർട്ട് അയച്ചു, അടുത്ത മാസം 100 അയച്ചു. By 3 months or 6 months, they will be having or they will be collating all this information, and they will be categorizing it. This particular drug is causing this much side effects. They will be categorizing based on the effect is caused. Cardiovascular effect aano, CNS effect aano, whatever the adverse event caused, they will be categorizing it. Based on that, they will be deciding the fate of that particular drug also. Okay, so we will go deep into that. So this is what I said on the ICSR. So you can see a physician or a patient or something like a clinical trials. These are the resources. We will be getting the reports. If I am having a drug which is manufactured by me, I am giving to the patient, I am responsible for reporting whatever the adverse events happen for that drug. Yellow card, you know yellow card, right? This is the same way. If I am getting an yellow card, I have to report it to whomever the manufacturer. Say for example, Cipla ano, Pfizer ano, whomever it may be. Cipla product in effect and dial, that particular effect should be monitored by Cipla. Okay, so we will be getting a receipt or a report. After that, it will be going to different entries, and there they will be having a designated database or a software. So we have to incorporate all this information appropriately into the database, designed database. Once it is done, the data entry will be completed. Now it will be going to medical review. What does it mean by medical review? Again, it is a group of physicians or doctors. So they will be analyzing whatever we have reported. ഇത് എൻ്റെ ഡ്രഗ് കൊണ്ടാണോ? ഇപ്പോൾ ഐ ആം ബീയിങ് എ മാനുഫാക്ചർ, ഞാനൊരു പ്രൊഡക്റ്റ് മാർക്കറ്റിൽ ഇറക്കിയാൽ ആ പ്രൊഡക്റ്റിൻ്റെ കംപ്ലീറ്റ് സേഫ്റ്റി എൻ്റെ ഉത്തരവാദിത്വം. സോ ഐ വിൽ ബി ഹാവിങ് എ ഗ്രൂപ്പ് ഓഫ് ഫിസിഷ്യൻ. ദേ ഹാവ് ടു അനലൈസ് എൻ്റെ ഡ്രഗ് കൊണ്ട് മാത്രമാണോ ഈ എഫക്റ്റ് ഉണ്ടായത്, ഓർ എനി അതർ റീസൺ ഇസ് ദെയർ ഫോർ ദാറ്റ്. സോ ദേ വിൽ ബി അനലൈസിങ് ആൻഡ് ദേ വിൽ ബി sending it for all the purpose, and finally it will be reaching to regulatory submissions. This is the workflow design for an ICSR, individual case safety reporting guidelines. Okay, now this just same thing whatever we are request, report collection from, so report collections, enquiries, literatures, whatever we are having, okay, all the resources we are having, and these are the basic elements of an ICSR. There should be an identifiable patient, suspect should be there, an event should be there, and the reporter should be there. Okay, now these are some terminologies in PV. MEDRA, there is some standardized coding guidance for MEDRA, that is called as MEDRA, that is Medical Dictionary for Regulatory Activities. Okay. So this is some example of MEDRA, that is we will be classifying it based on the reported event. So if you are taking rhinitis or running nose, again it will be going to a PT of influenza, we will be classifying it like that. And this is the reporting time frames. If it is a serious event, that should be submitted within 15, whatever we have deposited earlier, that should be submitted within 15 days. If it is a non-serious event, that should be submitted to the regulatory days within 30 days. Okay. So this is the causality assessment, again what we discussed earlier, whether that action or that event is occurred because of me or my drug. Okay. So we will be assessing it. There will be, you might have heard about Naranjo scales. Maybe in your textbook you might have seen that Naranjo scale, but that will be having all this information, certain, probable, possible, unlikely. But when it is come to the regulatory perspective, there is nothing like that, only binary. Either it is related or it is not related. This event, that is very simple. So binary assessment is only required for regulatory authorities. So these are some special situations, whatever we are basically focusing: pregnancy cases, pediatric and elderly population, vaccine cases. So this we already discussed on the risk factor part, why we need a pediatric and an elderly patient assessment. For assessing it more clear way, why it is pregnancy cases, we need some trials in pregnant patients to evaluate the effect in fetus, mother and father. If father is taking drug, whether it is affecting the fetus, mother is taking drug, whether it is affecting the fetus. All these aspects we need to review. And the vaccine cases, again you heard about COVID vaccines, right? Now update COVID vaccines are not effective, studies on complete. So we will be analyzing on that. Okay. So again coming to PBR, this I already explained. This is a periodic report. It will be happening based on the reports whatever we have shared. Again aggregate reporting is done by regulatory authorities. So this is happening. We are manufacturing drug undakiya. First 2 years, every 6 months they have to generate a report and analyze it. And again next 2 years, every year they have to analyze it. Next 3 years, every 3 years they have to analyze it. And they will be collating it. And what is the importance of this? If PEBR is analyzing this is not up to the market or not up to the expectation, they can make the drug to be banned from the market. Okay. So this is just an introduction, just the structure of the PEBR. This is the way it will be. Introduction, current, worldwide, update of actions, all those aspects. And it should be updated on this way only. Okay. So these are some new trends on social media cases. So if a drug is involved, if my drug is involved, definitely wherever being the report, social media lavam, Twitter lavam, Facebook lavam, are report either, that is case on. And the drug is case on. So I am responsible to report that to the stakeholders or regulatory authorities. Okay. And reactive, proactive, outcome oriented. How we are doing pharmacovigilance? Maybe on reactive approach. And the drug eduthu 100 report kitti. So based on that, ആ 100 report based I will be analyzing my drug. എന്തെങ്കിലും changes വരുത്തണോ? Whether I need to do some changes. Second thing, proactive. So how it would be? Maybe within ആദ്യത്തെ 2 report കിട്ടിയപ്പോൾ തന്നെ I have to analyze. ഇതിങ്ങനെ സംഭവിക്കുന്നുണ്ട്. So how I can prevent this in future? That way. Okay. And the last one is RPA, NLP, ML, DL and AI. Any idea? RPA is robotic process automation. NLP is natural language processing. ML is machine learning. DL is deep learning and AI. So as technology develops, we will be having all these into pharmacovigilance as well. Previously, നമ്മൾ ചെയ്തുകൊണ്ടിരുന്നതിനേക്കാൾ വ്യത്യാസമായിട്ടാണ് നമ്മൾ ഇപ്പോൾ ചെയ്യുന്നത്. Okay. So this is on the new trends. And again RPA and all, maybe Junaid sir will be explaining more on that. Okay, so you people will be getting to advanced on that. So RPA, NLP and all, these are the, if I am taking an example, മുമ്പൊക്കെ നമ്മൾ ഒരു കംപ്ലൈൻറ്റ് രജിസ്റ്റർ ചെയ്യണമെങ്കിൽ you have to call to the call center. Call center call ചെയ്ത് അവരുമായിട്ട് സംസാരിച്ച് you have to register it. അതിനുശേഷം we are having IVR, press 1, press 2, press 3, right? ഓരോന്ന് വിളിച്ച് ചോദിക്കുമ്പോൾ എന്താണ് കംപ്ലൈൻറ്റ്, 1 press ചെയ്യുക, 2 press ചെയ്യുക, 3 press ചെയ്യുക. Now no need for that. നിങ്ങൾ പറയുന്ന കംപ്ലൈൻറ്റ് അസസ് ചെയ്ത് തിരിച്ചു പറയാൻ ആൾക്കാരുണ്ട്. There is IVR is working in behind. That is automation, right? That will be happening here also. Okay, fine. So any questions? Sir.
Good afternoon, everybody. First, I would like to appreciate Mr. Manu, for handling a post-lunch section so beautifully. It was indeed very focused and pleasant. That is what I felt.
Thank you, sir.
I also appreciate the management, organizers for selecting the speakers from various areas. Why I said this is because when I noticed, when I was working outside Kerala and inside Kerala, what difference I felt was like in Kerala, students are more like academic oriented. They study well, they get good marks. But outside, I felt, my observation, students are more like they want to pass, but they are more job oriented. What all jobs they have, what is the other jobs I can have. They are very much aware of that. I think that area, our state is a little bit lagging behind. Of course, what I meant by this sentence is, other than the information he has given, hope all the students have thought about what is the speaker, what are the all speakers, not only Mr. Manu, all the speakers, what are their job profiles, what they are doing in the industry. Because I think most of the students will be concentrating on one or two jobs, which is very familiar. Apart from that, what all you can do, what all are there, they are real examples for that. You can feel real. Industry job sometimes feels very unreal for the students. They think it is just something which is heard, we have just heard about it. But you have real people coming from industry here, which of course you should be motivated about that. Again, you have more sessions which will again motivate you. Okay? And now the session is open. I think you have some questions on this, because definitely if you are thinking about something other than dispensing pharmacy and above that, you will definitely have some questions. So questions are welcome.
Yes, please, go ahead.
Good afternoon, sir.
Yes, go ahead.
Previously, sir was told about AI, how the processing was improved. I'm asking that how AI can improve risk assessment and adverse effect in the pharmacovigilance.
Again, AI, how can improve on the
Not only
pharmacovigilance?
AI, different technologies are developed currently. How different technologies can develop or advantages for risk assessment and adverse effect nowadays.
Again, so the adverse effects, whatever in pharmacovigilance I mean, okay, artificial intelligence is something like, so previously whenever we are doing case processing, we require to write everything manually. Okay, now when automation comes in or artificial intelligence comes in, എല്ലാം മെഷീൻ തന്നെ ചെയ്യുന്ന പ്രോസസ്സാണ്. സോ ഇഫ് എ റിപ്പോർട്ട് ഈസ് കമ്മിങ് ഇൻ, പ്രീവിയസ്ലി ഞങ്ങളത് മൊത്തം വായിച്ച് ഓരോന്നോരോന്ന് കണ്ടുപിടിച്ച് വി ഹാവ് ടു റൈറ്റ് ഇറ്റ് ഡൗൺ, മാനുവലി റൈറ്റ് ആൻഡ് നറേറ്റീവ്, എവറിത്തിങ് വി ഹാവ് ടു ഡു. പക്ഷേ ഇപ്പോൾ വെൻ ആർട്ടിഫിഷ്യൽ ഇൻ്റലിജൻസ് കംസ് ഇൻ, മോസ്റ്റ് ഓഫ് ദ തിങ്സ് ആർ ഓട്ടോമേറ്റഡ്. സിസ്റ്റം ക്യാൻ ഐഡൻറ്റിഫൈ എവറിത്തിങ് ആൻഡ് പുട്ട് ഇറ്റ് ഇൻ ദ പ്ലേസ്. ഒരു ഡേറ്റാബേസ് ഉണ്ടെങ്കിൽ ആ ഡേറ്റാബേസിൽ ഓട്ടോമാറ്റിക്കലി വെക്കാനുള്ള എല്ലാ സാധനവും സിസ്റ്റം തന്നെ ചെയ്യുന്നുണ്ട്. That is how AI is working in pharmacovigilance now. Maybe advanced things definitely will come. We can see for that. Okay. Yeah. Thanks for that.
Thank you, sir.
Sir,
That is. Yes,
Sir.
go ahead.
related to this AI, how reliable is the data which has been obtained from the AI? Like you said about data processing. In data processing, you use AI. So how reliable is the data which has been obtained?
Again, we can't completely rely on AI as well. So there will be definitely human intervention. ഞാൻ നേരത്തെ പറഞ്ഞതുപോലെ once the AI is completed also, there will be a QCer or a person who is reviewing all this data. But that will be limited. So AI can incorporate everything to the system, but there will be one person who will be verifying everything rather than an AI. So definitely human intervention will be there. Hope that can answer.
Thank you, sir.
Okay.
Okay, I think time is also, we are getting delayed. So I think at least one or two questions were there which was very relevant. It shows that some interest is there. When such a session happens, there is some interest among the students. So we would like to wind up the session. Thank you, sir.
As we conclude this session, we are taking a moment to present our speaker, Mr. Manu M., Pharmacovigilance Specialist, with a token of appreciation and our gratitude. For doing this honor, I invite Mr. Anthony Malikal, Secretary, Holy Grace Academy of Engineering and Holy Grace Polytechnic College onto the dais. Sir, please. Thank you, sir. It was an honor to have Dr. Anup KR, Principal, Triveni College of Pharmacy, for chairing the session. To present him with a token of gratitude, I invite Dr. Gio Baby, sir, Director, Holy Grace Academy of Management Studies, onto the dais. Sir, please.
Thank you, sir. Before the next session, delegates, please attention. For your kind information, we have arranged the transport facility to Thrissur route by our institution buses, which will start after the conference. So those who are interested to avail the facility can use it after the conference only.
As we continue to dwell into the theme of today's seminar, it's a time to move forward to our sixth session. This promises to be an engaging session shedding light on artificial intelligence and robotics in pharmacy. We have an excellent speaker lineup to provide us with a deep insight and thought-provoking ideas. We are privileged to invite Professor Junaid V, Vice Principal, Al-Shifa College of Pharmacy, for the session. For chairing the session is Dr. Sapna Srikumar, Principal, Ahalya College of Pharmacy. Both are requested to occupy the seats on the dais. We are fortunate to have Professor Dr. Sapna Srikumar, a distinguished academician, researcher and a leader in the field of pharmaceutical sciences. She serves as Principal, Ahalya College of Pharmacy, with over 28 years of experience in teaching and research. Her contributions to academic and pharmaceutical community have been truly remarkable. Ma'am is an accomplished researcher with 3 published patents, including innovations like modified drug release carrier for anti-cancer drugs. She has served several research grants from Department of Sciences and Technology, including a grant for natural peptide antimicrobial coats. Ma'am has also received numerous accolades, including the prestigious Women Achiever Award from INSO in 2022 and a State Government Women Excellence Award in 2021, recognizing her outstanding contributions to education and research. Beyond her academic contribution, she is an editor-in-chief of Annals of Pharmacognosy and has collaborated on several groundbreaking projects. Her commitment to mentoring the next generation is evident in her role as a PhD guide at multiple universities. She has successfully organized national and international conferences, published research, and fostered industry collaborations. It is an honor to have such an academician and visionary leader with us today. Please join me in welcoming Professor Dr. Sapna Srikumar to chair this session. Ma'am, please. Over to you, ma'am.
A very good afternoon to everybody gathered here, respected dignitaries and all the faculty members and dear students. Today, last but not the least, This is going to be a wonderful session by Dr. Junais. Junais sir is very efficient, not only in academics, administration, but also in various fields. I have seen him actively participating, and it is a real honor to chair your session, sir. And your topic on AI and robotics, which is the new trend and the latest trend, will help each and everyone. This is a avenue where everybody has to evade, because unless and until you are familiar with AI and robotics, then your future will be dim. So let us enjoy this session, and I welcome Dr. Junais sir to kindly take over.
Thank you, madam. Thank you very much. I'm sorry. I like to stand here. You have any problem? No. So guys, please stand up. If possible, give a shake hand or hug to the next person. If possible, give a shake hand or hug to the next person. Oh, you know why? You know why? The artificial intelligence cannot do this. This one can be done by only human beings. Keep a smile, hug each other, please sit. And I guarantee that this is an interactive session, and kindly occupy the front seat, please. I challenge you, kindly occupy the front seat. It's a challenge. It's a challenge. Challenging an X generation. Okay? With the Z generation. Balarama, Balamangalam, Mangalam, Vanida, Manorama. X generation childhood emotional factors. Maradona, Beckham, David Beckham, Steffi Graf, Gabriele Zabatini. These are our nostalgia. And in between us, there is Y generation. You know that 1980 to 96 is the Y generation. And you people are the last generation in an alphabet. Z generation. Yeah, 96 to 2012. Hope you are from 96 to 2012. Madam and all long back, I mean, First World War, Second World War generation. Leave it. Okay, anyway. And, ningal parayum, Tanda vibe undu parayula. You people are the next Tanda vibe, because Alpha generation from 2012 to 2025. And now, a new generation started from 2025, January 1st onwards. Can you guess? Beta. So for that people, you are a Tanda vibe or Talla vibe. Can I use dual language? ചിലതൊക്കെ മലയാളത്തിൽ പറഞ്ഞിട്ട് കാര്യമുള്ളൂ. തമിഴുണ്ടാ? ഉന്നോട് ഞാനിരുന്ന ഒവ്വൊരു മണിത്തുലിയും മരണപ്പെടുക്കലും മറക്കാതെ കൺമണിയെ കണ്ടതിരുക്കോലമെല്ലാം കനവാകം മറയുന്നാലും കടസിയിലെ എഴുത കണ്ണീർ കയ്യിലുണ്ട് ഒട്ടുതടി. So 2025, the AI challenge is nothing but attaching emotion with artificial intelligence. That's why I kept that figure. And have you seen simulations? No. Simulations. Illa. For your kind information, ഇവിടുത്തെ കോളേജുകാർ എന്നെ പറ്റിച്ചു. അവർ പറഞ്ഞു 3 മണി കഴിഞ്ഞാൽ എല്ലാവരും പോ. ഇവിടെ ആരും പോയില്ല. ഞാൻ അതിൻ്റെ ടെൻഷനില്ല. ഇത്തിരി ആൾക്കാരുണ്ടല്ലോ ക്ലാസ് എടുക്കാൻ. 4-5 ആളാണ് ഈ കാര്യം പറഞ്ഞു കൊടുത്ത് വിട്ടാൽ മതി. No one has gone. I think most of the people are here. So that's my tension. Okay? അവനും എന്നെ പറ്റിച്ചു. ഇത് ഞെക്കിയാൽ മതി എന്ന് പറഞ്ഞു. ഞങ്ങണില്ല. ആ ഓക്കെ, ഇത് വീഡിയോ റൺ ചെയ്യാൻ പറ്റുമോ? വീഡിയോ ദിസ് വീഡിയോ ആ യാ. This is a scenario without AI. Just enjoy it. ടിക്കറ്റ് എടുത്ത് കാണണ്ടല്ല. യു ആർ നോട്ട് ആക്സെപ്റ്റിംഗ് ചാലഞ്ച്. ഷെയിം. ഇങ്ങോട്ട് ഇരിക്കാം. ഞാൻ തിന്നൊന്നുമില്ല. കം, ബി കംഫർട്ടബിൾ, ബി കൂൾ. കിട്ടുമെങ്കിൽ മതി. നോ? Okay, no problem. So you know that the important thing is why they are sitting back, because it's a small criticism. AI and Z generation are very similar. You have one similarity. You know that AI and Z generation have some similarity. Okay. They are getting all the information. AI and Z generation are getting all the information from internet. And you people tell that I know everything. AI also telling that I know everything. You people also getting all the information from net, and you may tell that I know everything. This is a Tanda vibe with your generation. Clear? So coming to, HMPV. What is HMPV? As a pharmacist, you should know what is HMPV. What is that? ഒരു മൈക്കും കൂടെ തരോ, ഒരു ആൻസർ പറയണേ കൊടുക്കാൻ വേണ്ടിയിട്ട്. HMPV. First you should have self-respect. What is HMPV? Human meta? Yeah. Last 1 week, current trend HMPV. And this one and pandemic, COVID, that also, COVID well maintained by artificial intelligence, because, we have undergone so many diagnoses, disease diagnosis, and, Trump ഇപ്പോൾ കയറിയില്ലേ? Trump, last time, he was demanding something when COVID-19 was there. That is only because of artificial intelligence taught them. ഒരു സംഭവം കണ്ടുപിടിച്ചറിയാം, ഡ്രഗ് റീപ്രൊപ്പോസിങ്. കേട്ടിട്ടുണ്ടോ? ഇല്ല, സമാധാനം. ഡ്രഗ് റീപ്രൊപ്പോസിങ് എന്ന് പറഞ്ഞാൽ എന്താ? പാരസിറ്റമോൾ അല്ല, ചക്ക സീസണായാൽ നമ്മൾ എന്താ ചെയ്യുക? ചക്ക സാധാരണ കൂട്ടാൻ വെക്കും, തോരനാക്കും, ചോറല്ലാത്ത എല്ലാം ചക്കയാക്കി മാറ്റും. So suppose you are finding that paracetamol is useful for another disease, that is called repurposing. That is can easily get done by AI. Okay. So US artificial intelligence team told that one antimalarial drug can be used against COVID vaccine, COVID virus. ഏതായിരുന്നു അത്? Can you guess? Hydroxychloroquine, yeah. Wonderful answer. Another clap would get. So that same Trump demanded, and 1 or 2 ship full of hydroxychloroquine went to US only because of artificial intelligence. Okay, now we'll go to the session. So disease diagnosis, predictions, in which area, that the things will happen, and which state more predominant will be there. So that kind of things will be done by, in case of HMPV also, okay, treatment, tracing, everything is done by artificial intelligence. Okay, let me go into it. Sorry. Chata, ഇതൊന്നും റെഡി. സോറി, ഞാൻ ഇങ്ങോട്ട് അല്ലേ നോക്കണ്ടേ? ഫുൾ സ്ക്രീൻ. ഞാൻ തൊട്ട ഇടുക്കിയിലെ വരെ കറണ്ട് പോവില്ലേ? പണിയായ? അതെ, എന്തു പറ്റി? ഞാൻ റെഡി. ഇതെങ്ങനെ ചെയ്യാ? So what is AI, you know? What is AI, you know? What is AI? Artificial intelligence. It's like mimicking, simulating human brain. Mimicking or simulating human brain. Okay, you can human brain having this kind of efficiency, understanding, reasoning, solving problem, effective communications. Okay, and you know chatbots you are using, chatbots you are using. No, ChatGPT. ഇപ്പോൾ എത്രയാ വേർഷൻ? ChatGPT 5 is going to introduce by 2025. All are eagerly waiting for that. ChatGPT 5. Okay, so chatbots you know. I mean, എനിക്ക് കുറച്ച് ഫാസ്റ്റ് ആക്കാനുണ്ട്, അതുകൊണ്ട് നിങ്ങൾ പെട്ടെന്ന് പോകണ്ടേ? നിങ്ങൾക്ക് മെല്ലെ പോയാൽ മതിയോ? പെട്ടെന്ന് പോകണം, അപ്പോൾ ഞാൻ പെട്ടെന്ന് തീർക്കണം. ഓക്കെ, സോ AI can be, AI is the biggest umbrella. Okay, AI is the biggest umbrella. And with AI, you know, we are finding, solving problems, effective communication, reasoning. And under this umbrella, machine learning comes. What is machine learning? Is nothing but tracing datas. Okay. So let's example ornara. Bird. Okay. We are giving datas of pala type of birds, different birds. Yeah, kori, kaka, puchana, alla, kaka. പിന്നെ കുയിൽ, അങ്ങനെ 10 ബേർഡ്സ് ഡാറ്റാസ് വി ആർ ഗിവൺ. ലൈക്ക് ബേർഡ്സിന് എന്തൊക്കെയുള്ളത് പാർട്സ് പറ? നോർമലി ഇല്ല പാർട്സ്, ഡീപ്പായിട്ട് പോകണ്ട. ദാറ്റ് ഇസ് ഡീപ്പ് ലേണിംഗ്. എന്താ ഉള്ളത്? പീക്ക്, പിന്നെ വിങ്സ്, ഫെദർ, ലെഗ്സ്, ടോസ്. ആൻഡ് വി വിൽ ഗിവ് ഇമേജസ് ഓഫ് ഓൾ ദീസ് തിങ്സ് ആൻഡ് വിങ്സ്. ഓക്കെ, and we will give images of this one, okay, one by one, and that is basic data for machine learning. And finally we have given the details of 10 birds, like 20% foot, 20% feather, 40% beak, like that. നമ്മൾ 100% പോകുമ്പോൾ different proportions in different data is given. Clear? And finally, these 10 birds data stored in AI. Okay? And we are giving the photo of another bird. What will happen? The machine recognizes it as a bird. Okay? That is also based on the proportion. Suppose you are giving the photo of penguin as the 11th one. What will happen? Suppose the feather, the wings consist of 40%, and you are giving photo of penguin. What will happen? Penguin is a fish. No, bird. So what will happen? Machine may not recognize this one as a bird because its feather, its wings are not visible. Okay, so machine may tell that this is not a bird. Again you have to correct the machine. Like what you have to do? You have to give additional information that this is a bird, and machine will recognize. Like that, you also contributing. How? You people also contributing. How? എൻ്റെ തല, എൻ്റെ കുടൽ, എൻ്റെ full figure. Hundreds of photos you are taking and uploading. All these things will be utilized by AI team to recognize. Like അങ്ങനെയല്ലേ മെസ്സി മലപ്പുറത്ത് വന്ന് പന്ത് കളിച്ചു എന്ന് പറഞ്ഞിട്ട് ആർട്ടിഫിഷ്യൽ ഇൻ്റലിജൻസ് അല്ലേ? മെസ്സിയും ഒരു പുതിയാപ്പിള പോലെ നടന്ന് കല്യാണത്തിന് പോകണം. ഇറ്റ്സ് ആർട്ടിഫിഷ്യൽ ഇൻ്റലിജൻസ് ബിക്കോസ് സോ മെനി മലപ്പുറം ഫോട്ടോസ് ഓഫ് ഗിവൺ ടു ദറ്റ് ടീം ആൻഡ് ദറ്റ് ദേ ഹാവ് ഇൻകോർപ്പറേറ്റഡ് മെസ്സി. ദാറ്റ്സ് ഓൾ. So how much data we are giving, that much accuracy will come. So this is the case, machine learning, data, okay. And deep learning is nothing but is a neural network like our neurons, okay. This is a matter of nano level, microscopic, target level. We are targeting a particular helical structure of a DNA, okay. For that, we need millions of data. For machine learning, you need only thousands of data. But deep learning means millions of data and it will be accurate, targeted. So these are the basic structure of AI. First one is nothing but artificial intelligence, problem solving, communication, all these things, okay? And in that umbrella, machine learning is there. Thousands or lakhs of data will be collected, and with that data, predictions will happen. And the third one, it is nothing but deep learning. Deep learning, fine level, nano level, micro level, or lakhs or billions of data will be analyzed. Okay? For deep learning, more accuracy will be there. So this one, as I told, different patterns we will be uploading. Based on the patterns that algorithm workouts. Algorithm is nothing but step-by-step instructions. That happens in pandemic also, step-by-step instructions. നിങ്ങൾ അതിൽ കൊടുക്കുന്ന ഇൻസ്ട്രക്ഷൻസ് സ്റ്റെപ്പ് ബൈ സ്റ്റെപ്പ് ആയിട്ടായിരിക്കും. സോ ആ ഇൻ അൽഗോരിതം ബേസ് ചെയ്തിട്ട് AI will be created. Okay. So application, image processing, video processing, sentimental analysis, speech recognition, natural language processing, internet things. Okay. And history, a very short history. 1956, John McCarthy, he introduced the term AI. And 1964, first chatbot built, 1964. And 1997, you know that, have you heard about Deep Blue? Deep Blue? No, it's a computer, supercomputer that defeated chess player Gary Kasparov. The chess player Gary Kasparov, world champion Gary Kasparov. 2002, a vacuum cleaner learned to navigate independently. And 2011, Siri, you know that, iPhone with the Siri. Okay, that is introduced. And 2020, ChatGPT 3 introduced. And 2023, generative AI is nothing but total, from the images, new images will be created. That is called generative AI. Generative AI introduced. Okay. And 2024-25, there's now the what thing is happening means immense research is happening to make a emotional AI. Okay. So AI components, as I have already explained, okay. And generative AI, I told, generating new content. For that, you see, text, new content will be like generating AI use text messages, okay, note taking, general writing, support, sales, okay, a text message. Then coding will be there. AI supposed to take coding, okay, web app builders, text to SQL, code documentation, code generation, then image designs, media advertising, consumer social image generation, then voice synthesis, okay, then video editing, then 3D models, then music, RPA, gaming, everything will be explained, okay, biology and chemistry, okay. So 95% of AI industry, pharma industry is using, 95% of pharma industry is using AI, and it shortens the thing. നമ്മൾ പണ്ടൊക്കെ vaccine, it will take 12 years. Drug innovation also 6 to 10 years. Now the time is shortened within 1 year. That's why COVID vaccine happened within 1 year. Okay, so time shortened, and I mean for clinical trial also 70%. Clinical trial benefit anda. That's the importance of simulation. I will explain. Clinical trials nu parnanda. You have to do it in human, animal, okay. All these things can be time and everything can be saved. How it is saved, I will explain, okay. And minimum 26 billion saved per 4 years, okay. And these are the common AI tools. AI tools. You can, if you want, you can take a photo, okay. And these are the pharmaceutical industry AI tools. Generative, this is the RN and all neural level, deep learning, convolutional neural network. These are deep learning things. Deep Q networks also deep learning. Okay. And why AI? Why AI? I will explain examples only. Okay. I will make it fast. Okay. Identification of potential therapeutic targets. Traditional, in case of traditional, നമ്മൾ എന്താ ചെയ്യുക? ഒരു ഡ്രഗ് ബേസിക്കലി എന്താ ചെയ്യുക നമ്മൾ? അത് ബേസ് വെച്ചിട്ട് നമുക്ക് ഉള്ളിലേക്ക് പോകാം. എന്താ ചെയ്യുക? വാട്ട് വിൽ ഡു? നമ്മൾ സാധാരണ ഒരു പ്രോജക്റ്റ് എങ്ങനെ ചെയ്യണം? M.Pharm സ്റ്റുഡൻസ് ആർ ദ പ്രോജക്റ്റ്. How a project happens? B.Pharm final year. How a project happens? We will select one drug. Am I right? Am I right? We will select one drug and we will find out the shortfalls of existing treatment. We will find out the shortfalls of existing treatment. Suppose TB. TB existing shortfalls. What is the shortfalls? ഇത്രയും കാലമായിട്ട് ഐസോനിയാസിഡ്, റിഫാംബസിൻ, എത്താമറ്റോൾ, പൈറസിനമൈഡ് ഈ 4 ഉള്ളൂ. ഫസ്റ്റ് ലൈൻ ഡ്രഗ്സ്. And you are taking rifampicin, your urine will be like, cherry red color, cherry red color. Okay. And both are what? Controversial. Okay. That will happen. What catabolic, metabolic activity happens in liver. Both are controversial or opposite action. So liver get damaged. These are the basic problem. So what will you do when a drug formulation? You will go to library, you will find so many other solutions or new excipients, new things to be added with the drug. For that, it will take years. Am I right? You have to read all the books. Okay. Instead of that, you can do data mining with AI. What is that data mining? And then data mining. Mining no nanda. കോൾ മൈനിങ് ഒക്കെ ഇട്ടില്ല. നമ്മളെന്താ സാധാരണ ലൈബ്രറി പോകണം. ഇപ്പോൾ അൽഷിഫയിലെ ലൈബ്രറിയിലില്ല, ഹോളി ഗ്രേസിലെ ലൈബ്രറിയിലില്ല, ഇവിടെ ഇല്ല, ത്രിവേണിയിൽ ലൈബ്രറിയിലില്ല, എവിടെയില്ല. നമ്മൾ കുറച്ചും കൂടി അഡ്വാൻസ്ഡ് ലൈബ്രറി പോകും. അല്ലെങ്കിൽ നമ്മൾ ഇവിടെ പോകണം, എയിംസിൽ പോകണം, ഓക്കെ, ഡൽഹിയിൽ പോകണം, അല്ലെങ്കിൽ നമ്മൾ വേറെ രാജ്യത്ത് പോകണം ലൈബ്രറി. ഡാറ്റ മൈനിങ് എന്ന് പറഞ്ഞാൽ എന്താ? ഗെറ്റിംഗ് ഇൻഫർമേഷൻ അല്ലേ, ത്രൂ AI. അങ്ങനെ ലാക്സ് ഓഫ് ബുക്സ്, ലാക്സ് ഓഫ് റെഫറൻസസ്, ലാക്സ് ഓഫ് തിങ്സ് വിൽ ബി ദർ. അതിൻ്റെ ഓപ്റ്റിമൈസേഷനാണ്. സോ നമ്മൾ ഫസ്റ്റ് എന്താ ചെയ്യുക? നമ്മൾ ഡ്രഗ് എടുത്തു, എക്സിപ്പിയൻ്റ് എടുത്തു, ദെൻ ഫോർമുലേറ്റ് ചെയ്തു, ദെൻ ഇവാലുവേറ്റ് ചെയ്തു, ദെൻ പ്രൊഡക്റ്റ് ഇറക്കി, ദെൻ ക്ലിനിക്കൽ ട്രയൽ നടത്തി, ദെൻ വാട്ട് വിൽ യു ഡു? മാർക്കറ്റ് ചെയ്യും, ദെൻ മാർക്കറ്റ് സ്റ്റഡി. ഓക്കെ. ദിസ് ക്യാൻ ബി വെരി മച്ച് ഈസൺ ബൈ artificial intelligence. So it will take trial and error, so many trial and error, so many, it will be time consuming and costly, traditional method. But in case of artificial intelligence, see, AlphaFold provide highly accurate protein structure prediction. That will be very, very, very useful in cancer treatment. Protein structure and exact location kandupidikkum, site, X, Y, chromosome level also that will go. Okay, that's the importance of AI. And it is time consuming, already told. Example, during COVID-19, okay, AI-driven platforms like Benevolent AI identified baricitinib as a potential therapeutic in a matter of arthritis in a drug. Arthritis in a drug. AI koduthapo within weeks, it find out that it is useful for COVID-19. So, pandemic time urgency and emergency. So that time you need faster informations, you have to get very fast. So then a limited space exploration, as if you are a chemistry student, if you are studying chemistry, okay, you have to go to a chemical library for so many interactions. What is dating? Dating എന്താ? സാധാരണ കുട്ടികൾ ഡേറ്റിംഗ് ചെയ്യാറാണ് ആണോ അല്ല? സിമിലർ തന്നെ ഡോക്കിങ്. നമുക്ക് ഇഷ്ടപ്പെട്ട പാർട്ട്ണറിനെ തപ്പി പിടിച്ച് കൊണ്ടുവരുന്നതാണ് ഡോക്കിങ്. പെയറിനെ കൊണ്ടുവരുന്നതാണ് ഡോക്കിങ്. സിമ്പിളായിട്ട് പറയാം. അപ്പോൾ ഏതാണ്ടോ മാച്ച് ആവണ, നിങ്ങൾ ഇത് കണ്ടിട്ടുണ്ടോ? അവതാർ അല്ലേ? അവതാറിൽ ഒരു പക്ഷിയുണ്ട്, നമുക്ക് മാച്ചായ പക്ഷിക്ക് വാലിട്ട് കൊടുത്ത്, ദാറ്റ് ഈസ് കോൾഡ് ഡോക്കിങ്. സിമ്പിളായിട്ട് നമുക്ക് മനസ്സിലാവുന്ന ഭാഷ അത് അല്ലേ സാർ? ഡോക്കിങ്, ഡേറ്റിംഗ് ആൻഡ് എവറിഥിങ് ഈസ് സെയിം. യു ഗോട്ട് ഇറ്റ്? യാ. So, and lack of comprehensive data. I mean, altogether, combined data will not be available. For that, ഞാനല്ല കേട്ടോ, ഞാനല്ല. ആരെങ്കിലും വന്നാൽ നിങ്ങളും എന്നെ പ്രാഗാണ്, ഞാൻ പെട്ടെന്ന് തീർത്തരാം. ആ, നിങ്ങൾ ഓക്കെ ആണോ? ആണോ? നമ്മൾ പണ്ട് പിന്നെ ഞങ്ങൾക്കൊക്കെ ആനിമൽ എക്സ്പീരിയൻസ് ഉണ്ടായിരുന്നു. ഇപ്പോൾ ഫ്രോഗ്, മുയലൊക്കെ ഉണ്ട്. ഇപ്പോൾ മുയൽ പകുതി ചത്തിട്ടുണ്ടാവും. അപ്പോൾ അതിൽ കണ്ണിൽ ഇത് പിന്നെ എണ്ണയല്ല, മെഡിസിൻ ഒറ്റിച്ചിട്ട് കണ്ണ് ഡയലേറ്റ് ചെയ്യും, വലുതാക്കും. ഇപ്പോൾ എക്സാമിനർ വരുമ്പോൾ ഇത് ഡയലേറ്റ് ആവില്ല, ഇങ്ങനെ കിടക്കും, പീപ്പിൾ ഡയലേറ്റ് ആവില്ല. അപ്പോൾ നമ്മൾ സ്കെയിൽ വെച്ചിട്ട് തലമണ്ടയ്ക്ക് ഒന്ന് കൊടുക്കും, അപ്പോൾ ഡയലേറ്റ് ആവും. സിറ്റുവേഷൻ അതാണ് ഇപ്പോൾ, അതാണ് സിറ്റുവേഷൻ. ഓക്കെ, so comprehensive total data is AI-driven platforms like Tempus integrate patient genomic and clinical informations. Personalized data ക്ക് വേണ്ടി ഒരാളെ പേഴ്സണലൈസ്ഡ് ട്രീറ്റ്മെൻറ്റ്. ഹാവ് യു ഹേർഡ് അബൌട്ട് പേഴ്സണലൈസ്ഡ് ട്രീറ്റ്മെൻറ്റ്? ഇല്ല, ആണോ? നമ്മളിപ്പോൾ എന്താ ചെയ്യണേ? എല്ലാവർക്കും പാരസെറ്റമോൾ ആണോ? അല്ലേ? വൺ സൈസ് ഫിറ്റ് ടു ഓൾ, അതാണ് നമ്മളെ പോളിസി. പേഴ്സണലൈസ്ഡ് ട്രീറ്റ്മെൻറ്റ് എന്താ? നിങ്ങളുടെ റേസ് നോക്കും, നിങ്ങളുടെ ജീൻ നോക്കും, നിങ്ങളുടെ ഏജ് നോക്കും, ഓക്കെ, കപ്പാസിറ്റി നോക്കും. പൈസ കൊടുക്കാൻ വേണ്ടേ ബില്ല്? ഓക്കെ, പേഴ്സണലൈസ്ഡ് ട്രീറ്റ്മെൻറ്റ് ഈസ് ലൈക്ക് ദാറ്റ്. സോ പ്രഡിക്ഷൻ ആക്യുറസി നമുക്ക് പിന്നെ ട്രഡീഷണൽ മെത്തേഡിൽ ഹൗ ഇറ്റ് വർക്ക്സ് വി ഡോണ്ട് നോ എക്സാക്ട്ലി, ബട്ട് ഹിയർ വി ക്യാൻ AlphaFold predicts protein structure with more accurate identification of binding sites and interaction. ഒരു AI ജനറേറ്റഡ് ചാറ്റ് ബോട്ട് ഇതാണ്, AI ജനറേറ്റഡ് സിസ്റ്റമാണ്. ഇറ്റ് ക്യാൻ ഫൈൻഡ് എക്സാക്റ്റ് ബൈൻഡിങ് സൈറ്റ്. ഓക്കെ, ആൻഡ് ഇൻഎഫിഷ്യൻറ്റ് റീപർപ്പോസിങ് എക്സിസ്റ്റിംഗ് ഡ്രഗ്സ്. റീപ്രപ്പോസ് ചെയ്യുക എന്ന് പറഞ്ഞു കഴിഞ്ഞാൽ one drug can be used for another purpose. Okay, another repurposing. So see, അത് പിന്നെ കൺവെൻഷണൽ മെത്തേഡിൽ വെരി റെയർ. ആൻഡ് ഇൻ കേസ് ഓഫ് AI, so metformin is a diabetic drug. Metformin is a diabetic drug, and it found out that it can be used for cancer also. So drug repurposing. A lack of personalized, I already explained. And human simulations. What is human simulations? ഇതൊന്നും ആൾക്കാരല്ല കേട്ടോ. See, it is there in Kerala University of Health Sciences. Worth RS 1 crore human simulations are there. നിങ്ങൾ കണ്ടിട്ടുണ്ടാവും. ഇത് എന്തിലുണ്ടാക്കിയുള്ളത്? ഹ്യൂമൻ ബോഡീസ് തന്നെയാണ് അതേപോലെ ഉണ്ടാക്കിയത്. ഏത് വെച്ചിട്ടാ? സിലിക്കോൺ വെച്ചിട്ട് ക്രിയേറ്റ് ചെയ്ത സോ മെനി തിങ്സ് വിൽ ബി ദർ. ആൻഡ് ഇറ്റ് പ്രൊഡ്യൂസസ് ടിയേഴ്സ്. No, see, when we are giving data, okay, when we are giving disease condition, that body, it seems artificially, it sounds or mimicking the same conditions. If you are having cough, if you are giving cough as data, then it starts coughing, and if you sneeze, it has to sneeze. And delivery also, okay, without a human being, okay, the simulations can make birth of a child, but child also silicon. All things can be simulated. So human simulations, if possible, you can visit Kerala University of Health Sciences, and with the permissions letter. Okay, you have to give request. So AI mainly focused on virtual screening, clinical trial, predictive modeling, facilitating personalized medicine, optimization of drug formulation. These are the 5 major categories. Okay, 5 major categories: virtual screening, clinical trial, and predictive modeling, facilitating personalized medicine, optimization of drug formulation. I will make it fast. Okay, virtual screening is nothing but drug molecule versus target. Okay, because of data mining or virtual screening, because with that we can do drug molecules and target protein combination. And example, COVID time, main protease, okay, SARS protein, main protease MPRO, that is inhibited by, virus is inhibited by one genomic drug. Okay, so AI found out that, and it started blocking replications. Okay, so next, have you heard about de novo drug design? What is de novo? What is novel drug? New drug, advanced drug. If it is found out by AI, it is called de novo drugs. New drug, okay, novel drug. If it is found out by AI, it is called de novo, fresh drug. De novo means fresh, okay? And designing novel HIV protease inhibitors using molecular docking and AI. Clear? And repurposing, I have already explained. Synthetic route of prediction, in case of ibuprofen. Ibuprofen normally prepared with the complicated steps, and nowadays AI found out that it can be easily prepared, okay, by reducing the steps, and it's eco-friendly. And yesterday's, what is yesterday's, you know, news with CSIR, you know? What is yesterday's news with the CSIR? Don't know. Paracetamol also, okay, produced from India, or they found out that a very new method to produce pure paracetamol, okay, with the new technologies, that also by AI. So clinical trials, you know that so many stages of clinical trials are preclinical protocol, phase 1, phase 2, phase 3, phase 4. All areas, data, AI can be having a crucial role. Okay, and especially, I will give one example. Conducting a phase 3 clinical trial for a new diabetes drug, comparing its efficacy and safety against standard treatment in 10,000 patients globally. That word is more important. Globally. See, suppose you are doing that clinical study here, okay, you will get at least 10, minimum 10 or 20 patients with a similar gene. Okay, and with the AI, you will get as much as patients with a different race, age, everything. Okay, so this can be clinical trials can be improved. The efficacy can be improved by AI. Okay, and patient recruitment and selection, that also we can use AI. With the AI, you can set the age limit, and a particular age limit, you can include the patients. And adverse events and detection safety monitoring, that also, in case of vaccine trials, now so many complaints with the COVID-19 vaccines. So in case of vaccine trials, any complication comes, that also found out by AI. Okay. Then predictive modeling, you know that predictive modelings, by historical biological chemical data, you can find out what kind of things happens to a tumor, cancer tumor. Okay. So in what stage the tumor occurs. So data we are giving to the AI, and replication happens in same manner. Okay. And the time series forecasting, suppose this is the winter season. In this time, so many diseases will be there. So AI will tell you that in this month, what kind of drug is produced, and in this month, what kind of drug is marketed. Clear? And anomaly detection means any fraud, any untoward incidents happens, it's very easy to find out by AI. Anomaly detections, okay? Then customer behavior analysis. That is in pharmacy, and normally, YouTube on search okay. ഇപ്പോൾ നിങ്ങൾ ബോച്ചെ ആൻഡ് ഹണി റോസ് എല്ലാവരും ഉണർന്ന് സെർച്ച് ചെയ്ത് കഴിഞ്ഞാൽ എന്ത് പറ്റും? പിന്നെ അങ്ങോട്ട് ഫുള്ള് ബോച്ചെ റിപ്പോർട്ടർ, ബോച്ചെ 24 ന്യൂസ്, ബോച്ചെ മനോരമ, ബോച്ചെ കൈരളി, ബോച്ചെ. അതാണ് കസ്റ്റമർ ബിഹേവിയർ അനാലിസിസ്. ഓക്കെ. ആൻഡ് ഫെസിലിറ്റി പേഴ്സണലൈസ് മെഡിസിൻ പറഞ്ഞു, ജീനോമിക് അനാലിസിസ് പറഞ്ഞു, ഇമേജ് അനാലിസിസ്. ഓക്കെ, നമുക്ക് ഇമേജസ് ക്രിയേറ്റ് ചെയ്താക്കിയിട്ട് സ്കാനിങ് ആൻഡ് ഓൾ. ഇത് ഫാർമസി ഫീൽഡല്ല, സോ അതുകൊണ്ട് പറയാം സ്കാനിങ് അൽഗോരിതം എക്സാമിൻ MRI സ്കാൻസ് ഫോർ ഏർലി ഡിറ്റക്ഷൻ ഓഫ് ബ്രെയിൻ ട്യൂമേഴ്സ്. ഇമേജസ് കോൺട്രിബ്യൂട്ട് ചെയ്താൽ ഇമേജസ് ആക്യുറേറ്റ് ആയിട്ട് റീജനറേറ്റ് ചെയ്ത് കഴിഞ്ഞാൽ വി ക്യാൻ ഫൈൻഡ് ഔട്ട് ബ്രെയിൻ ട്യൂമർ ആണോ ഇതുള്ളത്. ഓക്കെ. ദെൻ റിമോട്ട് പേഷ്യൻറ്റ് മോണിറ്ററിങ് ബൈ മൊബൈൽ ആപ്പ് സെൻസേഴ്സ്, റിമോട്ട് പേഷ്യൻറ്റ് മോണിറ്ററിങ് വിൽ ഹാപ്പൻ. ഓക്കെ. Then patient engagement and education, lifestyle diseases, medication adherence. With the chatbots, a health chatbot, you can tell the patients how to change your lifestyle. Okay. Then optimizing drug formulation is nothing but total, drug formulation consists of drug, excipients, all the things. And for smooth functioning, that accuracy, that action, everything should be optimized. Okay. So one example is sensory optimization like chewable vitamin tablet adjusting flavor chewable vitamin tablets adjusting flavor. Okay. Chewable vitamin tablets adjusting flavor and texture, that is can be done by AI. Okay. And this cannot be run, I think so. And I will come to the next robotics very fast. Okay. So, have you heard about cobot? Cobot. ഇല്ല, നിങ്ങൾ ധൃതി കണ്ടിട്ടാണ് കസേരയിൽ നിന്ന് എണീറ്റ പോലെ ഇരിക്കുക. അതാ ഞാൻ സ്പീഡാക്കണം. ഓക്കെ, ടെൻഷനായി. റോബോട്ടിക്സ് പെട്ടെന്ന് തീർക്കാം. ക്ലിയർ ആണോ? കംഫർട്ടബിൾ ആണോ? നിങ്ങൾക്ക് വാട്ട് എ പ്ലാൻ മീൻസ് ഐ എം ഓപ്പണിങ് ഓൾ ദ വിൻഡോസ്. നിങ്ങൾക്ക് ഓരോ ഏരിയാസ് എക്സ്പ്ലോർ ചെയ്ത് തരാം. അല്ല, എനിക്ക് നോളജ് കാണിക്കാൻ വേണ്ടി ഏതെങ്കിലും ഒന്നെടുത്ത് ഭയങ്കരമായിട്ട് എക്സ്പ്ലെയിൻ ചെയ്യാം. നോട്ട് ലൈക്ക് ദാറ്റ്. നിങ്ങൾ B.Pharm M.Pharm സ്റ്റുഡൻസ് അല്ല, നിങ്ങൾ ഓരോ ഏരിയ കാണണം. അതിന് വേണ്ടിയിട്ടാണ്. ഓക്കെ. എന്താണ് റോബോട്ട്സിൻ്റെ സ്പെഷ്യാലിറ്റി? ഇപ്പോൾ ഇത് റോബോട്ട്സ് ആണെങ്കിൽ ഐ വിൽ ബി കംഫർട്ടബിൾ. എന്താ കാര്യം? റോബോട്ട്സ് ആണെങ്കിൽ എന്താണ് കാര്യം? ഐ വിൽ ബി കംഫർട്ടബിൾ. നിങ്ങൾ ക്രൗഡ് റോബോട്ട്സ് ആണെങ്കിൽ എന്താ കാര്യം? ഐ വിൽ ബി കംഫർട്ടബിൾ. വൈ? നമ്മൾ മലയാളികൾ വേറെ സ്റ്റേറ്റുകാരെ കുറ്റം പറയില്ലേ? ഞാൻ ആ വാക്ക് ഉപയോഗിക്കണില്ല. റോബോട്ട്സ് അല്ലേക്ക് നമ്മൾ ദുബായിലെ ഗൾഫിലെ ബംഗാളി ആരാ? മലയാളിയാണ്. ഇവിടുത്തെ ബംഗാളി ആരാ? ബംഗാളിയാണ്. അപ്പോൾ റോബോട്ട്സ് എന്താണ്? ദേ ആർ കോൾഡ് ബംഗാളീസ്. നിങ്ങൾ ഉപയോഗിക്കുന്ന വേർഡ് അങ്ങനെയാണ്. മലയാളീസിന് അങ്ങനെ ഉപയോഗിക്കേണ്ട, അത് പാടില്ല. ഞാൻ പറഞ്ഞുതരാം. എന്താ കാര്യം? ബസ്സിന് പോകണ്ട, ടയേർഡല്ല, ബിരിയാണി കഴിച്ചിട്ടില്ല, 24/7 നോ കംപ്ലൈൻറ്റ്സ് അല്ലേ? ദാറ്റ് ഇസ് എ മെയിൻ ബെനിഫിറ്റ്. ഒരു ടീച്ചറെ സംബന്ധിച്ചുള്ള റോബോട്ടാണെങ്കിൽ വി ക്യാൻ ടേക്ക് ക്ലാസ്. നമുക്ക് സമാധാനം. അതായത് നമുക്ക് ടീച്ചേഴ്സിനെ എങ്ങനെ വെച്ചാൽ ലോഡ് ചെയ്ത് വെച്ചിട്ടുണ്ടാവും. ഇത് എങ്ങനെ പറയാതിരിക്കുക ഭയങ്കര സങ്കടം. ഓക്കെ, ആ ഇൻഫർമേഷൻ ഞാൻ കുട്ടി കൊടുത്തു എന്നുള്ള സങ്കടം. അപ്പോൾ റോബോട്ടാണ് ഇരുന്ന് കേട്ടോളൂ. ക്ലിയർ? ഇപ്പോൾ സിനിമയുടെ ഒക്കെ ഇപ്പം തന്നെ എന്നെ തല്ലും, അതിന് റെഡി ആയിട്ട്. സോ റോബോട്ടിക്സ് ആക്യുറേറ്റ് ആണ്, എഫിഷ്യൻ്റ് ആണ്, 24/7 ആണ്, സേഫ് ആണ്. ലൈക്ക് എസ്പെഷ്യലി ഇൻ ഹസാർഡസ് എൻവിറോൺമെൻറ്റ്. സൾഫ്യൂരിക് ആസിഡ്, ഹൈഡ്രോക്ലോറിക് ആസിഡ്. ഹൈഡ്രോക്ലോറിക് എൻവിറോൺമെൻറ്റ് സ്പെഷ്യാലിറ്റി എന്താ? വാട്ട് ഇസ് ദ സ്പെഷ്യാലിറ്റി ഓഫ് ഹൈഡ്രോക്ലോറിക് ആസിഡ്? സോ പിക്ചേഴ്സ് ഉള്ളു. ഹൈഡ്രോക്ലോറിക് ആസിഡ് സ്പെഷ്യാലിറ്റി എന്താണ്? സ്പെഷ്യാലിറ്റി കുറച്ചൊന്ന് ഇൻട്രാക്ട് ചെയ്താൽ എന്താണ്? അതർ ആസിഡ് കമ്പയറിംഗ് വിത്ത് ഹൈഡ്രോക്ലോറിക് ആസിഡ്. ഹൈഡ്രോക്ലോറിക് ആസിഡ് ഇസ് എ ഗ്യാസ്. തുറന്നാൽ ഇവാപ്പറേറ്റ് ആവും. യു കനോട്ട് വർക്ക് അണ്ടർ ഹൈഡ്രോക്ലോറിക് ആസിഡ് വെൻ ഈസ് ഓപ്പൺ. ഓക്കെ, സോ ഇൻഹേൽ ചെയ്യും, ആസിഡിറ്റി കൂടും. ക്ലിയർ? സോ ഹൈഡ്രോക്ലോറിക് ആസിഡൊക്കെ hazardous chemicals to robots we can use. Okay, it's cost effective long term. History തുടങ്ങുകയാണെങ്കിൽ 1980s 1990. ഓക്കെ, ജസ്റ്റ് ഷോർട്ട് 80s നമ്മൾ ആദ്യം ഓട്ടോമേറ്റഡ് ഡിസ്പെൻസിങ് മെഷീൻ വന്നത് റോബോട്ട്സിൽ. 90s റോബോട്ടിക് പ്രിസ്ക്രിപ്ഷൻ വന്നു. ബാർ കോഡിംഗ് സിസ്റ്റം 90s തന്നെയാണ് വന്നത്. ബാർ കോഡിംഗ് സിസ്റ്റം അതൊക്കെ റോബോട്ടിൻ്റെ എന്താ പറയുക ഫസ്റ്റ് ജനറേഷനാണ്. ഓക്കെ, നെക്സ്റ്റ് ഇംപ്രൂവ്ഡ് റോബോട്ടിക് സിസ്റ്റം വന്നത് 2000 ഓട്ടോമേറ്റഡ് കോമ്പൗണ്ടിങ് വന്നത് 2000 2010 to 15 റോബോട്ടിക് ഫാർമസി സർവീസസ് വന്നു. AI ആൻഡ് മെഷീൻ ലേണിംഗ് വന്നു. റോബോട്ടിക് ആംസ് ആൻഡ് 10 to 15 വന്നത്. ഓക്കെ, ആൻഡ് 15 to 28 RPA സാറ് പറഞ്ഞ റോബോട്ടിക് പ്രോസസ് ഓട്ടോമേഷൻ വന്നു. 18 to 20 ഇൻവെൻട്രി മാനേജ്മെൻറ്റ് വന്നു. 19 പാൻഡമിക്കിന് വേണ്ടിയിട്ടുള്ള വന്നു. ദെൻ പ്രസൻറ്റ് ഡേ കോബോട്ട്സ്. വാട്ട് ഈസ് കോബോട്ട്സ്? കോബോട്ട്സ് എന്താ? കോബോട്ട്സ് അതായത് ഇപ്പം ഞാൻ എന്താ ചോദിച്ചതെന്ന് കുട്ടിക്ക് അറിയില്ല. ഷീ ഗേവ് ദ മൊബൈൽ. ഓക്കെ, കോബോട്ട് ഈസ് കൊളാബറേറ്റീവ് റോബോട്ട്. ഷീ ആക്റ്റഡ് ലൈക്ക് എ റോബോട്ട്. ഷീ ഡിഡ്ന്റ് ആസ്ക് മീ what you want. She just simply gave that. So Cobot, while you are doing the things, while doing the things, they will support collaboration with a human being. Cobots. So benefits of robots ഞാൻ പറഞ്ഞു കഴിഞ്ഞു. ഓക്കെ, ടൈപ്സ് ആണ് ഫാർമ ഫീൽഡിലുള്ളത്. വൺ ഈസ് ഓട്ടോമേറ്റഡ് ഡിസ്പെൻസിങ്. ഞാൻ പിക്ചർ ഇത് കിടക്കണില്ല, പിക്ചർ മാത്രമേ കാണിക്കുന്നുള്ളൂ. ഓട്ടോമേറ്റഡ് ഡിസ്പെൻസിങ് ക്യാബിനറ്റ്. ദിസ് മച്ച് ഡ്രഗ് in a patient, this kind of patient that will be dispensed by automatic dispensing cabinet. Okay, then robotic prescription dispensing system. Robotic prescriptions, based on the prescription that will take the medicine and that will distribute. Robotic prescription dispensing system. Okay. Next one is automated compounding system. Like sterile products. നിങ്ങൾക്ക് sterile product ഉണ്ടാക്കണം. സ്റ്റാർട്ടിങ് തൊട്ട് ഫിനിഷിങ് വരെ ഓട്ടോമേറ്റഡ് ആയിട്ട് അതിൻ്റെ പ്രോസസ്സ് കംപ്ലീറ്റ് കൺട്രോൾ ആവും. ഹൈലി സ്റ്റീറൈൽ ആയിരിക്കും. നോ കണ്ടാമിനേഷൻ, നോ ഹ്യൂമൻ ടച്ച്. Then mobile robots, this one is a real factor in we're in Singapore. എന്താണ് മൊബൈൽ റോബോട്ട് ഫോർ മെഡിക്കേഷൻ ഡെലിവറി. മെഡിക്കേഷൻ കൊടുക്കുന്നു, അത് പേഷ്യൻ്റ് പിന്നെ അത് ഫ്ലോറിലൂടെ പോകുന്നു. ദാറ്റ് വിൽ ഗോ ടു ദാറ്റ് പർട്ടിക്കുലർ റൂം ആൻഡ് ദാറ്റ് വിൽ ഡിസ്പെൻസ് ദ മെഡിസിൻ ആൻഡ് വിൽ കം ബാക്ക്. ക്ലിയർ? മൊബൈൽ റോബോട്ട് ഫോർ മെഡിക്കേഷൻ ഡെലിവറി. നിങ്ങൾ തീയിൽ ചവിട്ടി നിൽക്കണോണ്ടാണ് ഞാൻ സ്പീഡാക്കുന്നത്. ദെൻ റോബോട്ടിക് ടെക്നോളജീസ് ഇൻ ഫാർമസ്യൂട്ടിക്കൽ മാർക്കറ്റിംഗ്. മാർക്കറ്റിങ്ങിൽ എങ്ങനെയാണുള്ളത് നോക്കാം. മാനുഫാക്ചറിങ് നിങ്ങൾക്കറിയാം. ഇതിൽ വെയർ ഹൗസിലൊക്കെ നിങ്ങളെ കൊണ്ടുപോയിട്ട് കഴിഞ്ഞാൽ ഫസ്റ്റിൽ ഗൾഫിൽ ബീഫാമോ കഴിഞ്ഞ് പോകുമ്പോൾ മിക്കവാറും വെയർ ഹൗസിലാണ് പണി. ഓക്കെ, നമ്മൾ പറയും, പുറത്തുനിന്ന് പറയുമ്പോൾ ആൾക്കാർ പറയും ഗൾഫിൽ വലിയ ആളാണ് പറയും അല്ലേ. വെയർ ഹൗസിൽ പാക്കറ്റ് എടുത്ത് അങ്ങോട്ടും ഇങ്ങോട്ടും ഷിഫ്റ്റിംഗ് ആവും. കുറച്ച് കഴിയുമ്പോൾ നിങ്ങൾ ടയേർഡാവും. ഓക്കെ, ഫോർ ദിസ് കേസ് ഇറ്റ്സ് വെരി ഈസി. 24/7. സോ ഓട്ടോമേറ്റഡ് ഗൈഡഡ് വെഹിക്കിൾസ്. Okay, robotic process automation. I told total process will be controlled by robotic process automation. And robotic arms, that is selective, and they will take that things and it will go to, a particular function. Particular task will be given to that. Okay, and cobots. This is the total area of production. Production, research, development, quality and packing, finished product. This is the total process. And cobots with the human, that simultaneous synchronized work of human and robotics. Okay, cobots. That is called a collaborative robots. Then packaging and labeling. Labeling nowadays, prescription labeling accuracy will be there. നമ്മൾ ഹ്യൂമൻ ആണെങ്കിൽ കുറച്ച് സ്റ്റിക്കറൊക്കെ ഒട്ടിച്ച് പറിച്ചു പോകും. ഓക്കെ, ചിലപ്പോൾ സ്റ്റിക്കർ ഒട്ടിട്ടുണ്ടാവില്ല. അത് ആക്യുറസി 99% ആണ്. Okay, sticker, labeling, everything, packing and labeling robots. Okay, and labeling process another. Okay, and automated storage and retrieval system. This is the biggest challenge in factories. Okay, we have to keep one patch of drug in a particular area, and later in next day we will get confused with other areas. Okay, നമുക്ക് എവിടെ ഡ്രഗ് വെച്ചെന്നുള്ള കൺഫ്യൂഷൻ ഉണ്ടാവും. But in case of this one robot, it will happen very easily. Thank you. Thank God. എനിക്ക് കിട്ടിയത് 3:15 3:20 4:06 ഞാൻ ഓടിച്ചെത്തിയിട്ടുണ്ട്. ഓക്കെ, ഐ തിങ്ക് ഓക്കെ. ആൻഡ് ഐ തിങ്ക് നിങ്ങൾക്കൊന്ന് ഒരു റിലാക്സ്ഡ് ആയിട്ടൊന്ന് ഒരു ചെറിയൊരു ആക്ടിവിറ്റി സാർ, ചെറിയൊരു ആക്ടിവിറ്റി. നമുക്കൊന്ന് മഴ പെയ്ച്ചാലോ? ഒന്ന് റിലാക്സ്ഡ് ആയിക്കോട്ടെ. ഓക്കെ, ഒന്ന് മഴ പെയ്ച്ചാലോ? എൻ്റെ പരിപാടി
it was a very smart, very energetic, and very scintillating session. We have to give another kayyadi to sir. Junais sir, whatever Junais sir has taken today, delivered today, it was not that, it didn't seem like as if he's delivering a lecture. It seemed as if he's giving a movie, you know, like a 3D, 4D movie. Am I right? So it was excellent, very well done with a lot of effort, and I think nobody will forget this conference because of Junais sir's energetic approaches. And thank you so much, sir. Please understand that human beings are important, but artificial intelligence, learning about robotics, because that is the future generation, the alpha and the beta generation, as sir said. So keep it in mind and move forward. I wish you all the best. Any questions? Please come forward. Anybody with questions? Okay, so I think everybody has understood. No questions? All right, so thank you. Thank you very much to the management, principal, director, all the team, organizing team of Holy Grace, and everybody for such a beautiful day. Thank you.
It was an outbreaking session. Thank you, sir, once again. Here we have end of the session, and we are taking a moment to present our speaker, Professor Junais, with a token of appreciation and our gratitude. For doing so, I invite Mr. Jose Kanampalli, Sir Chairman, Holy Grace Academy of Pharmacy, onto the dais. Thank you, sir. It was an honor to have Dr. Sapna Sri Kumar, Principal, Ahalya College of Pharmacy, for chairing the session. To present her with a token of gratitude, I invite Mrs. Ramya Benny, CEO, Holy Grace Group of Institution. Ma'am, please.
Thank you, ma'am. And that's how we end the Hapscon 2025 International Conference on Advancements in the Global Pharma Arena. Let us now formally commence the valedictory section with a warm welcome to our dignitaries on the dais. So to formally extend a warm welcome to this valedictory function, let me invite Mrs. Lakshmi O.S., Associate Professor, Department of Pharmacology, Holy Grace Academy of Pharmacy. Please, ma'am. Delegates, attention please. We have the bus that has been arranged in the morning session as the same route to Thrissur. So those who are availing the same bus route can meet the main entrance at the Holy Grace Academy of Pharmacy after the conference. The same bus route is availing after the conference.
A very warm good evening to one and all gathered here. Esteemed guests, respected speakers, distinguished participants from various colleges, and dear colleagues. It gives great pleasure and immense gratitude that I welcome you all to the valedictory function of Hapscon 2025. On behalf of Holy Grace Academy of Pharmacy, first I welcome all the eminent speakers of this Hapscon 2025 to this valedictory function even in their absence. Next I welcome Professor Junais B, Vice Principal Al-Shifa College of Pharmacy for this valedictory function, and the chairperson of this today's session, Dr. Anup K.R., Principal, Triveni Institute of Pharmacy for this valedictory function of Hapscon 2025. Then I welcome Dr. Sapna Madam, Sapna Sri Kumar, Principal, Ahalya School of Pharmacy for this valedictory function of Hapscon 2025. Then I welcome all the Holy Grace management team, Mr. Sani Adatugarin, Chairman, Holy Grace Group of Institutions, Mr. Jose Kannambili, Chairman, Holy Grace Academy of Pharmacy, Mr. Benny John Ainikel, General Secretary, Holy Grace Group of Institutions, and all the management team, even in their absence. I welcome all to this valedictory function. Next, I welcome Dr. Ajit Kumar, Academic Director, Holy Grace Academy of Pharmacy for this valedictory function. Then I extend my welcome to all the chairpersons, all the principals from various institutions, and all the delegates to this valedictory function of Hapscon 2025. Once again, welcome you all. Welcome. Thank you.
Thank you, ma'am. We are now distributing the participation certificate for various colleges. Representatives from each college can come and collect the certificates of participation onto the dais. To begin with, I invite Mr. Sujit Sebastian Matthew, Vice Principal, Holy Grace Academy of Pharmacy, to hand over the certificate to the representative of Ahalya College of Pharmacy, Puducherry, Palakkad.
Next, I invite Alex Martin, Professor and HOD Department of Pharmaceutical Chemistry, to hand over the certificates to Chemist College of Pharmaceutical Sciences and Research, Ernakulam. Chemist College of Pharmaceutical Sciences and Research, Ernakulam.
Next, to hand over the certificates to the representative of Indira Gandhi Institute of Pharmaceutical Sciences, Perumbavoor, I invite Dr. Muthuramu Sir, HOD and Professor, Department of Pharmacology, Holy Grace Academy of Pharmacy. Please, sir.
I invite Karpagam College of Pharmacy, Coimbatore, to give away the certificates. I call upon Delphina CP, Associate Professor, Department of Pharmacy Practice.
To hand over the certificates to Nehru College of Pharmacy, Pambadi representative, I invite Mrs. Dishamol CP, Associate Professor, Department of Pharmaceutics, Holy Grace Academy of Pharmacy.
I invite Amida Soni, Assistant Professor, Department of Pharmaceutical Chemistry, to hand over the certificates to Nirmala College of Health Sciences, Chalakudi.
To hand over the certificates to Nirmala College of Pharmacy, Muvajapura, I invite Ms. Aishwarya Jit, Assistant Professor, Department of Pharmaceutics, Holy Grace Academy of Pharmacy. To hand over the certificates to the representative of Triveni Institute of Pharmacy, I invite Ms. Reshma Mohan, Assistant Professor, Department of Pharmaceutics, Holy Grace Academy of Pharmacy.
invite Krishna Priya TS, Assistant Professor, Department of Pharmacology, to give away the certificates to representative of Lissi College of Pharmacy, Ernakulam. Lissi College of Pharmacy, Ernakulam. Any Okay. So I invite IPA members to take away the certificates from Krishna Priya TS, Assistant Professor, Department of Pharmacology. IPA.
That comes to the end, and now as we approach the conclusion of this wonderful event, it is time to express our gratitude to all those who made this occasion possible. To deliver the vote of thanks, I would like to invite Mrs. Winnie Wilson, Assistant Professor, Department of Pharmaceutics, Holy Grace Academy of Pharmacy. Please, ma'am.
Respected and most invited guests, dignitaries on and off the dais, good afternoon to all. I deem it a great honor and privilege to propose the vote of thanks on this memorable occasion. Let me first of all start by giving glory to Almighty God for making today's occasion a resounding success. First and foremost, I express my sincere gratitude to Professor Dr. Jay Shekhar, President IPA Kerala branch, for accepting our invitation and spending her valuable time with us. Thank you, sir. On behalf of Holy Grace Academy of Pharmacy, I extend my heartfelt thanks to all speakers, Dr. Erin Lingenfelter, Clinical Pharmacist, University of Utah Hospital, USC, Dr. Venkatesh, Research Associate Professor, University of Utah Hospital, USC, Dr. Rajan Radhakrishnan, Professor, Dubai Healthcare City, Dr. L. Ramanathan, Associate Vice President, Technical Operations, Apex Laboratories Private Limited, Mr. Manu M., Pharmacovigilance Service Specialist, Assenger Pharma Limited, Bangalore, Professor Junais V., Vice Principal Al-Shifa College of Pharmacy, Perindalmanna, for gracing this conference and sharing with us your findings, opinions during this memorable and successful event. We have been fortunate to have some of the eminent personalities from the academia as our chairpersons. On behalf of Hapscon 2025, I express my sincere gratitude to Dr. Renjish, Assistant Professor, College of Pharmaceutical Sciences, Government Medical College, Alapura, Professor Dr. L. Sudarshan, Principal, Nirmala College of Health Sciences, Chalakudi, Dr. Anil Babu, Principal, Westford College of Pharmacy, Thrissur, Professor Dr. Shyam Kumar, Principal, Al-Azhar College of Pharmacy, Dr. Anup K.R., Principal, Triveni College of Pharmacy, Dr. Sapna Srikumar, Principal, Ahalya School of Pharmacy. I am thankful for all the delegates from different pharmacy colleges, especially Ahalya School of Pharmacy, Chemist College of Pharmacy, KMP College, Karpakam College of Pharmacy, Sanjo College of Pharmacy, Lissi College of Pharmacy, Triveni Institute of Pharmacy, Nehru College of Pharmacy, Nirmala College of Health Sciences, Nirmala College of Pharmacy, Muatura, Westford College, for coming to this Hapscon 2025 International Conference. I am sure that the participants must have been benefited by attending this conference. We are grateful to our management team. Without their generous support, it would not have been possible to organize this conference. I express my sincere thanks to Mr. Sani Adatugaran, Chairman, Holy Grace Group of Institutions, Mr. Jose Kannambili, Chairman, Holy Grace Academy of Pharmacy, Mr. Benny John Aynikel, General Secretary, Holy Grace Group of Institution, and all other directed board members for your support and encouragement. I express my sincere gratitude to all principals from different colleges for accepting our invitation and sending students to make this conference a grand success. Thank you all. I thank all the distinguished invitees present here for accepting our invitation. I thank all the principals and staff members from our sister concern for your valuable presence. My heartfelt thanks to our director, the organizing committee, heads of various departments and faculties for their valuable contribution, guidance and encouragement in all our efforts. I owe special gratitude to non-teaching staffs who have worked hard to make this occasion a memorable one. We have been fortunate enough to be supported and motivated by the team. I thank the volunteering committee and the good hearts who work behind the scene. Even if I missed anyone, I didn't mention their names. Thank you all. So once again, I express my thanks to all the team for making this event a grand success. Thank you.
As we conclude, let's all rise for the national anthem.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
With this, officially we conclude Hapscon 2024. Thank you all again, and have a safe journey back.